Hi everyone, welcome to Dive 790 with the ROV SuBastian. We are on the Climate Connections at the Icy Interface Cruise, and we are doing our last dive in the general area where there was a recent exposure of seafloor. We're outside that area, but in the same general area in the southeastern end of the Ronne Entrance off the Bellinghausen Sea. My name is Erin Easton. I'm an assistant professor at the University of Texas, Rio Grande Valley. I'm here with partial support of Ocean Census, and this cruise is part of the Challenger 150 initiative. Today in the room we have piloting Sunny and Andy, with Andy flying. We've got Carlos taking care of streaming, and we have Sveta and Anna in the room as well. And we are going to be transecting to our first waypoint here with minimal stops, but then after that we'll be doing some transecting mixed with faunal sampling. Today we're still targeting mollusks, polychaetes, paracarpods, corals, sea cucumbers, and of course we'll be doing some push coring later in the dive. This dive estimate time is around 16 hours. About 14 and a half of which is bottom time. So we'll come off bottom roughly in 14 and a half hours from now. So, Anna, Sveta.
Hi everyone. So as Erin just said, today in the room we have Erin, aka Professor Coral. We have Anna, Professor Seawater, and I'm Dr. Extruder. We're going to take you on a ride for the next little while.
Yeah, one thing you'll have to look forward to later in the day is we have a drill in about 7 hours from now or so, 7.5 hours from now. So that'll be fun. There'll be a nice loud noise coming through the stream and then waking everyone up on the boat that's sleeping. No, it looks like they have a lot of buffering. It is not the last dive, it's just the last dive in this area. We have one more station after this to go to. And those of us in the room are really happy about this last station that we're going to, because it's on the shelf of Antarctica where it goes deeper. So I don't think we're going to be diving much deeper. We're going to be diving in a similar depth, but we expect to see a lot of changes with the community out there compared to what we've been seeing on these other dives. So yeah, it'll be really exciting to do, and we'll be going to that station on our way out and back towards Punta Arenas, Chile. So yeah, we just sat down here on the seafloor recently, and you can see it's sedimented with plenty of sea pigs. I've seen some scallops. Looks like there's at least 2 species of sea pigs around. Some crinoids.
Yeah, I think our station at the outer shelf, so that is essentially where the kind of shelf seas ends and the open water deep ocean starts. And what happens is basically we think that all of this inner shelf area, like where we are right now, would have been completely covered by the ice sheets during the last ice age, or the last glacial maximum, which was around 20, or to be more precise, 23,000 years ago. And since then, the ice shelf retreated from the area and exposed the seafloor, and it continues to retreat to the current day. So yes, it's exciting. You get a lot of sedimentation on that sort of shelf break where the outer shelf, where it changes into the open ocean, and a very steep grade and depth transect. So we go from shallow areas, which we've been exploring here, between 400 to 1,000 meters approximately, and it gets very steep very quickly, and down to a few thousand meters, or around 3 kilometers depth. So as Erin said, hopefully we see very different fauna. I'm very interested in the sediments, very excited to collect some sediments from that sort of area. We're going to deploy some gravity cores as well. And we might even risk a 6-meter gravity core and risk both of our barrels. So that would be exciting, cover the whole deck.
Oh, that'd be nice. It'd be really interesting. How Do you have any sense yet of how far back in time the sediment goes, or you're not going to know till it's back in the lab?
Yeah, I won't know, but the stuff on the outer shelf from my previous experience working on sediments around here, and also specifically in Amundsen Sea, which is just around the corner, that sort of outer shelf can give you a really high resolution of the whole deglaciation. So as you went from the previous ice age around 23,000 years ago to the modern warm conditions 10,000 years ago, it gives you a really nice chronological geological sequence, so you can really investigate changes in this ocean-based sort of biogeochemistry, ocean circulation changes that affected this area, and how the ice sheets influence that through whilst deglaciating. So for me, I'm really happy we're going to station there as well, and not just all of this in the shelf ones, even though these ones have been extremely interesting.
Yeah. So do you know, did they end up deploying the float today after the CTD?
No, that's tomorrow.
They're going to do it tomorrow.
So we were meant to do another gravity core this evening, and have this dive slightly later. But basically then there was loads of fog. So we were meant to transit for about an hour to the other gravity core location and then transit back here and do the dive. But because of the fog, the captain said it'll take about 4 hours to get to the gravity core location. So then Sasha and Patricia, who are our chief scientists, got woken up to discuss. So we decided to stay in this area and not waste time transiting. So we're going to do this gravity core after the dive tomorrow, just before the floats and stuff. And that will be after that. There'll be the float deployment and collection of the gliders late into the night.
Okay, so yeah, after doing the float deployment, that will be another CTD ROV.
I don't know if it's with the float, but I think where they're recovering the gliders, they will be doing CTD, and if the gliders are in a different place, then I think they're hoping to do 2 ROV CTDs. So, but yes, they will need all eyes on deck, because when you collect these gliders, so one of them is being picked up from a small boat, the other one, you basically have to watch it as if it's a whale, and get very excited and try and spot the glider. Professor Fish, if he's watching, please do send comments about the fish. So
Yeah, especially if you want one.
Professor Fish will be joining you shortly in about an hour time for his shift, so I'm sure you'll hear a lot more about fish from the professor.
Yeah, we can keep going. Yeah, still in kind of transect mode till waypoint 2, so thank you. Yeah, as another one of the things, you go to, it's a matter of hours, the whole schedule changes, so.
Did you ever go to sleep? Because I saw you
No.
said you were going to sleep, so that was lucky I texted you before you went to sleep.
Yeah, I was having a hard time falling asleep,
know,
and I
I'm
saw
sorry.
the text, so.
I was gonna wake you up, but then I thought I'll let Erin deal with the wake-up calls. I already had enough of waking Sasha up, and then Patricia. They were not happy.
I can imagine.
Because I think both of them just went to bed maybe an hour before the wake-up call.
Yeah, we heard the phone ring, so we heard Sasha's phone ring.
Oh, that was me
door.
phoning him, yeah. And then I left him to go wake up Patricia, because I was like, okay. He was like, really, Mom, do I have to come there? And I'm like, yes, you do. Come to the mess right now. We're waiting for you. The captain wants answers.
Right now.
Right now, like she was, yeah. And then he was just like, and then just hung up. And then I was like, is he coming? Is he not coming? We give him 5 minutes, and then I'm going into the cabin.
Yeah, I thought maybe it was for missing gravity core waypoints again or something.
No, luckily not this time.
Oh, this is kind.
You know, the cruises, I think I was only woken up, I was woken up a couple times all in the same night, but it was because we had dead sub and not because there was something.
To tell the captain the coordinates of your next location.
Yeah. Or because we had to change something.
But I suppose here, because of the weather and all the iceberg
Yeah.
situation, is much more variable than your classic cruise planning.
Yeah.
Some beautiful rocks going on here that I'm just logging. So in the middle, the big one is a bit of exposed bedrock. So that's the ocean bottom sticking out essentially, with some sediment scattered around it. And then the big other individual rocks you see are probably some drop stones, which is when these ice sheets retreat, they spit out these drop stones as they move away from the area.
Yeah, and then we're seeing sea stars and anemones and brittle stars. There's a cup coral coming up on one of these rocks. So again, you see a lot of these filter feeders that crinoid up at the top of the screen that will attach to and hang out on these raised surfaces relative to the seafloor because the current's a little bit faster, so they can get a little more food.
Oh, nice zoom in on the rocks. Thank you very much. I love a good rock observation. Professor Rock will be happy in the morning. Professor Rock said I'm very good at logging all the rocks for him to look at later. Apparently on the last cruise there wasn't many geologists, so he was basically awake almost all the time for all the dives because he really wanted to log all the rocks.
Wow, that would be exhausting.
Absolutely. This is very beautiful.
It's very pretty.
Is this the type of coral you're interested in, Erin?
This is an anemone.
Oh.
Yeah. So
Whoops.
related. Related.
I thought it was the mushroom coral.
No, the mushroom coral. Actually, there might have been a mushroom coral on the one side there. Wasn't 100% sure.
Okay.
No, we're good. We can keep going. It's kind of in an awkward spot to collect anyway if it was one.
So what is this anemone thing? Is that an animal?
Yes.
Does it have like polyps like a coral, or does it have algae, or it doesn't?
It's like a solitary. It's essentially a solitary polyp.
Okay.
In the middle, there are tentacles in around, including some of them. That was secret.
Yep. Look at all these sea cucumbers again. The last few dives have just been loaded with sea cucumbers in some areas. It's impressive.
Is this not normally the case? Because I'm starting to feel like you just see them everywhere when you go to the seafloor.
Yeah, I mean, it's when it's sedimented, yeah, they're pretty common, but this density's on the high end of what I've personally seen. Not that I haven't seen it high before or similar densities, but we just on multiple of these dives now we've seen quite high density, so that's pretty impressive. Usually you see it here and there, but in this general area, I guess with all the increased productivity this time of year, they plankton bloom, and apparently so do the sea cucumbers. So it'd be interesting to see what the seafloor here looks like when it's covered with ice in the winter, which we need to send an AUV or something down to get that.
But I think for that you need an icebreaker as well, right? It's much harder to get into the area, like in winter. Plus, imagine how cold it would be on deck, like working with the samples.
Yeah.
I mean,
yeah.
it's already pretty chilly here in the summer, but no thank you. That's what I would
I
say
know,
to that.
right?
Because right now we're processing most of our samples and sediments in the hangar, science hangar. So this area is a bit warmer, is unheated, but it's covered. So it's kind of bearable if you wear a few layers. However, if you were on deck, like some days are better than others. So sometimes it's sunny and it's fairly warm and it's alright. Other days, like today, it's snowing, it's very cold, there's wind. You know, you go for a cigarette, your hand drops off after 5 minutes. So
Don't go for a cigarette.
I go for loads of cigarettes all the time.
There's some tunicates, it looks like here.
At least I get loads of fresh air. You know, some people just sit on the ship. They never need to go outside. And now I'm getting fresh air every few hours. It's great. Sometimes more often.
Yeah, we've had an awful lot of days where the science party is not allowed out on the deck. Whenever we have snow and it's icy conditions out, or if it's windy, the seas, because we've been inside this bay, have been quite calm. Even the day that we had like 36 knot winds or so, like we were barely rolling. And this ship is a rolly ship. It rolls like so easy. So yeah, it's been by far the calmest research expedition I've ever been on in my life. So yeah, other than the like 2 days of passing the Drake Passage on the way in, it's been pretty impressively calm, but we still, if it's high wind or it's snowing, we can't go outside, so we're kind of stuck inside.
We'll see what the Drake is like on the way back.
I know, right?
I'm getting excited.
Yeah. Yeah, me too. It's like countdown time, you know? Counting down to the last dives.
I mean, I've been really enjoying it, but I am also getting quite tired of processing all the samples. I think the first couple of days of transit, I'm probably just going to be like asleep. You shall not see me.
Yeah, I just like it when we have the sunny days. It's so nice. Like we'll have a couple of sunny days, and then we have these couple of gray, foggy days, and then the sun comes back out, and those days are really nice.
So beautiful, right? The sunny days.
Yeah. And then was it yesterday when we had all the whales around?
Yes,
that was really
yesterday
amazing.
evening, that was amazing. We were whale watching off the deck.
I think last night we saw pilot whales, and we also saw like a minke whale, I think. And then
Yeah, so-
this morning, I think they were seeing minke whales.
So okay, what's this say? Because we saw me too.
Yeah.
What
so-
time was that, Anna?
Yesterday morning.
Today? I feel like it's today. I haven't slept yet, so I don't know. Is that-
It's
is-
today? Today, yeah, sorry. Yeah, because my computer said that today is the 1st of February.
I mean, yeah, who knows? So are you guys going to sleep after the shift?
Yeah, until
I hope.
until about a half an hour or so before the drill.
Oh no.
No, the drill is at 10:30.
Yeah.
The drill.
So I'll sleep until like 10 or so.
I forgot the drill.
Yeah.
Oh no.
Well, I actually don't have to go to the drill because that's right in the middle of my like sleep time. So
Yeah, well,
David,
it's
excuse me.
right near the end of my sleep time, but it's gonna wake me up anyway, so it doesn't really matter.
I don't know. I
might
reckon
as well get
I'll
up and do
report
it.
tomorrow, but I reckon I'll be able to sleep through it.
Okay.
I probably won't wake up. You know, you've got to think about my evacuation alarms. Normally it takes about 5 before I actually even open an eye.
That would be amazing if I still slept like that, but it doesn't work for me that way anymore. When I was younger, it did. Not
This is what
anymore.
happens when you function on little sleep. Just sleep like a log.
Alright, so we're getting close to waypoint 2, and then after that we'll be able to stop and do more zooms and do some more collecting.
What are we collecting at the next waypoint?
Whatever we want to collect.
Yeah.
We've got the luxury of all the boxes being empty, so
Yeah, it's like our...
Erin, go wild, go to town, go shopping.
Yeah, let's
Get all
hope there's
the things
something
you want.
interesting.
I can. Okay, so you come
Fill up all the boxes on our shift. Sorry.
Can you imagine?
Yeah.
We finish everything.
I actually, on all of my other expeditions, I had a reputation for loading the box up, and like, especially when I was using the suction sample, I'd be like, oh yeah, suction that and that and that and that and that and that. Oh yeah, keep suctioning all in that same container in the one spot. So.
After 2 waypoints is done.
Yeah, there was one time we settled down on this huge coral forest, and every time we looked around, we can go and zoom in and sit down and look at this Dumbo. We're close enough to waypoint 2. Yeah, and it was so many species we hadn't seen yet, and we're just like, and collect that, and collect that. We spent like an hour and a half or 2 hours collecting before we ever made it anywhere. It was amazing. I think that was about how long it was. A really long time though. It might have been a little less.
This is a nice octopus.
Mm-hmm. Oh, the sleeping Dumbos. I don't think I've seen a Dumbo awake yet on this cruise.
Maybe it's
Yes,
the
the report-
top.
Yesterday, yes.
You did? Okay.
Yesterday morning. we
Okay.
saw it.
I slept through the only one we saw that was
I
awake.
will show you.
Yeah, like I've seen the other octopus awake, but not the Dumbo.
Okay.
Oh,
Finally.
so cute.
He's,
So interesting on this dive, we haven't seen any sponges yet because you know at the end of last dive there was so many.
look at how it's swimming.
Yes, I am on the next expedition as well. I don't know. Sometimes I wonder why I do this, but I love it. So, but yeah, 2 expeditions in a row again. So I'll be off the ship for 4 days and then come back on for another
That's so cute.
like 38 days, 35
Wow.
at sea.
Erin loves a good cruise.
I do.
Have you got any more planned after the next one this year, or is that it? for a
No,
while?
no, I don't. But you never know. Surprises. Like this next expedition wasn't one I'd originally planned either. So,
How'd you manage to get on all these cruises? There's so many in a row. That's crazy.
yeah. Well, I was the chief or co-chief on the first 2 that we did. The third one, Schmidt Ocean Institute invited us back when they needed a last-minute science party last year. Like they invited us 11 days before we had to board the ship.
Oh wow.
And we said, sure. Yeah, it was interesting. I was actually in Chile picking up samples from our prior 2 expeditions. So I was with my collaborator there when we got the word. And then I was flying back to the US when we were having meetings with SOI and Ocean Census about supporting us and getting us there and what we needed, what we could and couldn't bring. So yeah, that's how the third one happened for last year. And then I was a co-PI on another one last year, but didn't go on it. I just participated from home because I had to teach at some point last year. And then I was a co-PI on this one as well. And then the next one, they had open berths. And I thought, well, why not? It sounds like an amazing cruise, and I'm already going to be here. So,
How did you find out about it, that they had open berths in the next one?
so I'm on their
Watch
their
list.
alliance. Yeah, so I get like an email. Yeah. So they sent out email invitations, and I was actually at a conference. No, more like a workshop, like on seamounts, talking about policy and presenting on some of our cruises from last year, when a Chilean student that had been with me and also applied for the position was accepted to go on to it. And so, and he was the one who'd also forwarded me the thing and said, hey, I want to apply to this. Will you apply? Are you going to apply as well? And I was like, I don't know. I got to think about it. But I did. And so he was invited on like a Friday, maybe Thursday or Friday. And I didn't get an invitation. So I was like, oh, okay, I wasn't selected, but that's fine. I'd rather have the student go. And then Monday, I went into work and I had an email waiting for me. So, yeah.
Nice.
So I get invited as well. So he and I are both going. So,
So maybe if I do some more core extruding, they'll invite me back as well.
yeah, maybe.
The resident doctor extruder is needed on board.
Definitely.
Very possible. I mean, the SOI expedition last year, they invited 3 mappers and 8 innate member biology team. 7 of us were like part of our kind of team, my collaborators team. 7 of them, yeah, 6 were from my Chilean collaborators, students and researchers, and my collaborator, who's a professor, and then myself for the 7th person from our team. And then they also invited another Chilean biologist that hadn't been part of our cruises, but was part of another cruise. And so there were 8 of us total for the biology team to run 24-hour ops. And yeah.
It's a bit like on this ship, we have 5 geologists
Mhm.
running all the operations. It is quite hardcore, I have to say. Next time I go, I'd quite like to bring like a student assistant, you know, to just help do all the extruding. I mean, obviously I'd be involved too, but I keep having to ask people for help, you know, just whoever's around, because it's kind of the amount of work we're trying to do is too much for a team of 5 as well, to be fair. But so far I'm quite impressed with managing how much stuff we've managed to do despite that, so it's good.
Yeah, it's always amazing how much you can get done with a small number of team members. So, yeah, I don't know. Like on this next expedition, I'm not quite sure of the breakdown of the team members, but we have an awful lot of media coming on, taking the berths. So
Why is
I
that?
think, yeah, I think they're gonna be. I don't know. It's not all the details. I really don't know yet. So,
I guess you'll find out soon.
yeah, I'll find out more soon. We had an initial meeting and they introduced us to some of the. Just gonna do some extra filming on top of what SOI already does, from what I understand, and so, yeah, that'll be interesting. And then there's the biology team. They're splitting us into 2 teams to run the 24-hour shifts, but I'm not quite sure of exactly how many of us there are that are biology versus mappers versus other teams. So I think they sent me a document yesterday. So once we have a transit day, I'll look at it. After the meeting, they sent it. So.
That is a pink
Nice
rock.
rock.
Pink rock.
Pink rock.
Yep. Can we collect?
Oh yeah,
that
we're at the rock
Probably.
collection area, right? So
Yeah. You can collect that one. You do the.
maybe, guys, we should just fill all the biology boxes and have shift with rocks. I don't think people are going to be too happy when they wake up if that happens.
We also use suction for the rocks.
Bridge, ROV, can we pause the move to collect a rock, please?
Just one more.
Amic. You can read that?
Bridge, ROV.
Hello.
Hey, Phil, is there a way if we pause the move to collect a rock?
Yeah, totally.
All right, thanks.
Yeah.
It's just
Yeah, no, we do have time. I just want to make sure that we don't commit to 20, 30 minutes in one location without checking first.
Copy that.
Vessel stuff.
Can we get a nice picture of the rock, please, as well? Before we... Thank you.
So
Excellent.
does that look like a good rock?
Looks like a nice rock. Sure, Professor Rock will be
Where would
happy.
you like it?
Let's put in that outboard one.
Okay.
Box 1.
Here, we'll come up out of the
UTC 621. Alright. We can go back to transiting, and I'm gonna let Anna let you know if we wanna, or Sveta will let you know if we wanna zoom in on anything. I'll be back up at 5.
Okay.
I'm gonna clean up my laundry.
Oh? Okay, I'll keep an eye out for rocks. Anna, you have to do biology.
Oh, no way. I can't. Just let me do it.
Bridge, ROV, it says something move again.
On the move.
So yes, the pink rocks. So we see a lot of these darker black rocks. These are probably metamorphic metasandstones. Whilst these pink ones, we are thinking that they might be igneous and dropstones from the glacier. Alternatively, it could also be a coating overgrowth on the black metamorphic rock, so something like manganese perhaps. But we shall have a better look at this rock and maybe saw it in half once we get it back on deck. It's really cool. We've got a rock saw on the deck, and Professor Rock has a lot of daily fun sewing through these rocks. I just saw, Anna, you've sent Professor Rock an update that we've collected him a nice rock. That's great.
Yes. So when he wakes
he sees the nice rocks. He said we collecting the best rocks on our shift. Apart from his own shift, we're good at rock collecting.
Yeah, we are indeed. Oh, the pink rock, we collect all the pink rock.
Oh yes. Is this one, Anna? Do you want to examine this one? No.
Is an anemone is like the solitary solid, as we explained before. And
Wait, they can't really hear you.
And you.
Oh,
you
Aaron
can't
is
hear?
back.
Okay. I have this book.
Alright, I'm back.
Okay, I'm back. So,
That
Sorry,
was not
everyone,
5
I
minutes,
just-
that was like 1 minute, 30 seconds, very rapid.
you
Yeah.
Like
ran.
I didn't run.
I
It's
couldn't
just
even say this is an anemone. and
like an
here it came back.
ultrasonic jet to the laundry room and back, straight into the ROV control room.
well, it's pretty easy when you just have to pile the few clothes you have from one bin to the other.
I know, I have some stuff that I can't tumble dry, and we don't really have anywhere to hang it on the ship, so because I've been doing night shifts, I just hang it all over the computer room. Apologies to anyone who's seen clothes around the computer room. So this is when my laundry time is normally a good 3-4 a.m. around this time.
It is a good time. They were like all empty.
Yeah.
Actually, I have to say on this cruise, like, there's been very little competition for the washers and dryers.
I'm quite surprised. Everyone
Me
gets so
too.
muddy and has so much like of this all these thermals and stuff. I don't know why they don't do the washing.
I don't know either. I've never.
Maybe we should tell Jen to do a better cabin inspection
I don't know.
next week.
And lab inspection. I wash my clothes.
Are we going
I'm
upslope?
using for water sample, even though I have sediment on my pants. And they say,
Oh.
why? If I'm sampling water.
I remember back when
That's
I.
true, yeah. Why do you have sediment on you if you're just doing the water?
Sediment everywhere.
Sediment is everywhere. I'm sorry, Dr. Extruder's really getting greasy and out of control.
I mean, I get my clothes dirty just getting the samples out of the ROV because there's so much mud on the ROV. It's like
Ah, yeah,
it's
yeah,
squeezing
that's
around
where
it.
it's coming from.
Yeah.
So,
But I don't know. You know when I used to do coring, like my first cruise when I did coring with a multiple core, and I mean I had like 2 outfits that were like my mud outfits that I wore in the cold room when we were processing the sediment. But I had to do laundry like all the time because you'd wear those for like 3 days and it was just coated in mud, and I would just wash that separate from like the rest of my clothes.
yeah, this is what I'm doing with that boiler suit that I wear for extruding, because that just gets like covered. Do a couple, 2-3 dives and then just wash it separately.
Yeah.
Have we found any fossils inside the rocks? Someone's asking. So, no, but they are of a different nature. To find fossils, you need sedimentary rocks normally, not metamorphic or igneous. So igneous is made by like lava eruptions and magma and that kind of thing, whilst the metamorphic rocks are made under really high pressure systems. So, for example, plate tectonics, earthquakes, that kind of thing. We have, however, so my particular interest are these microscopic shells in sediments, which is why I'm after the sediments. So I'm a geochemist that studies these shells. They're called foraminifera. They're single cell marine protists. They're around the size of grain. Around the size of sand grain. And these shells are quite rare in Antarctica, but so far we've been really lucky finding quite a lot of them. So I've been very happy. Actually, really surpassed my expectations of what we might find. Now this is a nice juicy rock.
Yeah, it is. It's a big rock. Got an urchin wearing a nice size shell. Got
So yes, if you Google foraminifera, so it's spelled like Foraminifera, like you'll find they come in all shapes and sizes. And they're very pretty, even though they're small. So we have 2 microscopes on board, so I've been kind of preliminary doing a little bit of sieving of some scrap samples from the sediments we've been getting, and checking sort of every sample, seeing what's in there. Last night I had a few hours off, if you like, or didn't have any tasks, so spent a bit of time at the microscope, and basically I have this very thin paintbrush. if you think it's the thinnest one you can buy in an art shop, and you pick out, you sort of wet the tip of the paintbrush and you touch the shell out of all the rocks and sand grains that you see through the microscope, and it sticks to the wet brush, and then you put it in the microscope slide. I can show you
One
later,
by one, yes.
one by one. So yes, last night I've picked out some shells. So I've been dyeing them with pink dye, and there's some really nice, cool pink ones
Yeah, I
to show
wanna
you
see it.
tomorrow. Oh, maybe I should do some photos for the science meeting just
Yes,
to show a couple
Yeah,
tomorrow
that'd be really nice.
of what we've been finding. It's very interesting because a lot of them are from subglacial sediments from that sub-ice area we were sampling from the station 16, which I really wasn't expecting to see any there, because in theory they only live in open marine conditions, so I'll have to do some thinking, but some were pink, and essentially the way that this biological dye works is that the ones that are pink were alive at the time of collection, whilst the ones that are white, which is their normal colour, they were already dead. So yeah, I took this first sample yesterday and had a little play with the microscope, and was really surprised there's a lot of like very pink ones, so
Can you
they were
zoom in on
definitely
that? I
alive.
think this is a recently crushed urchin.
Oh, what would have crushed it? That looks quite sinister.
I think, because I've been seeing these little puddles of like pink, but this is the first time I've seen. But maybe it's something else. I don't know. Oh no, it's like sponge covered or sponge encrusted.
I don't know. It's so
Actually, I have a stupid question for biologists.
Yeah.
When these organisms die, so you know, normally on land they kind of.
Yeah, can we stop and look at it? I'm curious what it is.
So on land, like when things die, they kind of disintegrate in sediment, right? Like with worms, and you have like degradation.
Yeah.
Does that happen
What
on the seafloor as well?
This might be sealed.
So like the cucumber
Oh,
dies,
really?
and then it just
Oh, you
like,
saw
rots
something
away
earlier?
on the seafloor.
Oh, okay.
There is that, like,
Yesterday we saw it.
yeah.
Okay.
But we haven't really seen any dead
Okay.
ones.
No, we need that.
Well, we can zoom again. Okay, maybe.
It's so
Alright. Maybe.
maybe we can.
Alright, time to go on.
Fill up
Oh,
a
it's
suction
outside.
sampler with
Oh,
dead
you
things,
see,
Erin.
there is also,
Yeah.
Got it.
what is that?
We can put it in like section 3?
Sure. Three.
3, yeah.
Why 3?
They're different size meshes.
Oh, okay.
So I don't want to use the finest mesh ones in case they want to suction something really
Some
small.
prawns for Patricia.
Some
prawns.
Yeah.
You're learning very quick.
I'm running quick.
But this is a pretty small sea spider, so I don't want to use the regular mesh. So, no.
Yeah, I got it.
How does she preserve them? Does she just add ethanol to them?
I don't know if she's been doing ethanol or formalin actually. Yeah. But you know, when you were talking, I'm wondering if you can talk to Soph tomorrow. If you
Mhm.
take images with the,
The
or if
forams?
it's a feed with the forams, that she can show it during shift tomorrow during the dive with the live stream up in the little corner.
Oh, I see. To say, yeah, to that side, not to fear from science party.
Yeah.
Yeah, that could be quite cool, just
Well,
to
like,
show
show
what
what's
we've been
going on.
finding. Yeah.
But I think they could probably do that. She just has to say like that screen. You know, 'cause you were showing your camera the microscope the other day.
And
Yeah, I'll
you
try and get
in
that.
our
Spider.
shift, you can then explain.
Yeah.
Well, I don't know if I'll be here for our shift
Oh,
tomorrow.
yeah. And if
Okay.
not, why not? We
Because
will
I
be
don't do
here.
daytime shifts. You should know this. You
But
have Rachel.
tomorrow will be
Are you guys
special
saying you
day.
don't like having Rachel as my substitute? You
Yes,
miss
we
me.
like, but tomorrow is a special day.
You can just pop in at the end and talk about it.
That's true.
You
I
have to
might
be
be
there whole
just
shift,
about awake
like-
at the end.
Like 3:30.
Stuff as
Yeah,
well?
like-
Like today.
You can get a little bit of the potential
Oh,
seal
can we
feel.
please get like that
Yeah,
together with the.
Just a touch, because you might very well be right, which is weird.
I have a feeling the whole thing is.
There
gonna
we go.
suck
That's
it.
enough. Right there. That's
Okay.
good. That's enough. UTC
It is a spider.
632. Well, you know, we just want to give it some food. You know,
Number
the
3,
next.
right?
Yeah, number 3.
It's probably going to want to keep eating while it's in the suction sample dry. Alright, I wasn't watching to see if it went in, I'm assuming.
Yeah.
It did? Awesome, thank you.
Is
Yep,
it
I see
Anna?
him.
I need your help. Where is it
Perfect,
on
thank
this?
you. So,
That's it on the section.
Oh, the change system.
Oh,
And food unknown.
Wanna leave it on?
Yeah.
I'll add
Oh, you
hungry sea spider with its dinner leftovers.
Hold on, 3. Yep.
amazing. So, onward we go.
Anna.
Higher density of
And
these sea
if you
pigs
check
again.
Sophia's spreadsheet, she leaves all my comments in there, so it's great.
I don't know what depth,
Oh, that is
I wonder what depth she wants the sea cucumbers from this time.
Does it say?
No.
I'm sure she'll wake up soon and tell us.
She said she was gonna sleep though today.
I think she went from the
Oh, it says the shallow. It's towards the end there,
Awesome,
at
thank
waypoint
you.
12.
because
Okay,
yesterday
perfect, thank you.
from the deep.
That's awesome. Yeah, let's give this a really nice zoom. The laser's on it first, and then you can turn off the lasers and
Anna.
get some.
As I've just found in here also the Niskin bottle.
Flag it for
Yeah,
Alex.
You cancel.
Yeah, yeah, this is the system.
Oh wow.
And,
This is a nice fat sea star. I
Not
like to
all
imagine
fat
that
sea
it contains a lot of these sea
Oops,
cucumbers
and the
inside of it.
sediment
That it had a
storm
nice
came.
burger with pickles
Yeah,
for
sorry.
dinner.
Maybe we'll see it, how it's clearing out today. All right, good to know. We don't have to get the sea cucumbers quite yet.
It's a shame no one on board wants to collect this nice big fat sea star. I'd love to like have a poke of it.
very big.
What kind of
Very.
texture does it have?
Mm.
Do you guys know? How does it feel if you like touch it?
This one I would think is probably like harder to the touch, and then it has a lot of texture, so it's gonna be bumpy.
So is it like still jelly-like or like a tough jelly?
No, it's like tough. I would say this one's probably a tough one. There are ones that are like jelly-like, but they're called slime stars, and they look like slime stars. So like when you're looking at them, they're kind of poofy and pillow-like. So, but most are like harder.
I'm starting to get really hungry. It's approaching my third dinner time, which is at 4 a.m. If you guys would like to join me for my third dinner today.
For our first breakfast.
See, I haven't gone to sleep yet, so I had my, I don't know, third dinner or third meal of the day just before coming in.
Oh, I see. That's wise.
Yeah.
You couldn't.
Because you see, the problem is I wake up for dinner, so I have dinner for breakfast, so that's my first dinner. Then I have second dinner at around midnight, and then I have a third dinner at about 4 o'clock in the morning.
Nice.
Which is why they're not breakfast, lunch, and dinner, because I basically eat the same thing. 3 dinners.
Yeah, I tend to eat just lunch and dinner and then like a snack before bed. It kind of depends on when I worked out that day, or if I worked out.
This is a very large sea star. It's super cool.
Yeah, I mean that's it's over a half a meter for
Probably
sure.
half a meter, you think?
Yeah, the lasers
The
are 10.
lasers, yeah. Wow.
I'd say it's close to 60 centimeters.
So it's almost the size of my gravity core.
There's another sea spider floating through.
Spider over there. You see it?
Oh yeah.
Very tight.
It got suspended. Yeah, we've been seeing some of the sea star, but they've been coming in different color variations. I don't know if that's all the same species. It's been kind of a cream color, this orangish color. There was one we saw earlier that in addition to like the black bands near the tips, it had some black stripes like between the arms, or like a one vertical band between each of the arms. So yeah, don't know enough about this particular group to know if that's color variation or if there's different species within the same genus here. But yeah, I guess it's not quite
Back in 2 minutes.
the rock. And there we go, starting to clear up. Alright, can we zoom in a little bit on the texture? Wow. That's cool. And then maybe pan over to the foot. Alright, thank you. Yeah, we can go ahead and zoom back out and then head on our way. Yeah, it's huge. What now?
You can close one this.
Alright, can we close one Niskin bottle before we leave?
Thank you.
UTC 642. 40 meters. Alright, so we're just going to collect a Niskin bottle here for the eDNA. eDNA for this one?
Oh, shall I unlock
Yes.
this one? Have you locked it yet? No?
No.
Okay. What number is it?
One, but still, yeah.
Awesome, thank you.
Thank you.
Alright, onward we go. We're making some pretty good time here, so let's see. Oh no, I guess we're only coming up on waypoint 3, so yeah, still collections through waypoint 4, so we're good. Awkwardly swimming, sea cucumber. Yeah, it's at least 60 centimeters across.
Turn this stuff off, it keeps coming back on. Is that you?
No,
What's coming back on?
Adjusted camera and then it self-adjusted.
Hmm. So yeah, we're just gonna continue here. We're nearing waypoint 3, and then we'll make a slight turn off to basically the left, the port side, and keep moving up slope. Started around 800 meters.
So I guess these first few waypoints we just do like video transects mainly. Is that what they want?
Well, they said from like starting at 2, we could do. It was a potentially interesting area for faunal sampling, but it's. And we saw the sea spider, but we haven't seen anything else really on the.
No, but 3
the
doesn't
target
have
list.
any like comments attached.
Because
Yeah.
it's written until waypoint 4.
Like 2 and 3, yeah, 2 and like 3. You just
Oh,
follow
yeah,
2
those, yeah.
basically. So we have like an hour to get from 2 to 4, essentially, is the time they estimate. So yeah, in theory, they should get to waypoint 4 within in the next half hour. So I don't know
and
how
it says
long
rock
the
collection.
waypoints are from one to the other, though.
I might stay for a bit of the next shift because they want to collect rocks and they don't have a geologist, so maybe they could do some. Although so far we've only just seen really sediment, we've not had much geological. It's really interesting when we were on that sea mountain earlier. Actually, it was quite a lot of. I just logged quite a lot of geology things. In the previous dive this
Mhm.
afternoon.
And then,
Yeah.
this month, honey,
Yeah, it was. It's been, I think, really interesting geologically. In terms of fauna, I feel like, we've been seeing a lot of the same fauna. Their distribution differs from dive to dive and with depth, as my impression. We'd have to do, stats would have to be done later and more analyses
Log
before
it.
that, but we've been seeing a lot of the same species.
Do you normally, on other dives, like on cruises in different other areas, would you see more diversity in like different dives of fauna, or is this like standard?
I tend to see more diversity on different dives around the different features because it's very much depends on the current and the topography, and you tend to see areas with less sedimentation and other areas with more sedimentation. I see, like here we're seeing a lot more sedimentation, and that tends, in my experience, to lead to a little more similarity in at least the megafauna that you're seeing. There might be differences in fauna, but yeah.
But do you think, like in Antarctica generally, I would imagine it's less diverse, or at least say for my forams, like you have less species that are able to tolerate these kind of really cold, extreme environments? Is that the same for this kind of other deep sea fauna, like the mega and the meiofauna, or not so much?
I think it has like pockets of really high biodiversity, but yeah, there's groups that don't tolerate it well. We like squat lobsters aren't known from polar regions apparently. So, and that is a group that we would see really high diversity of at seamounts. So in non-polar regions at least. So yeah, I think it's a little bit of a mix. Even on the seamounts that we studied on the sedimented areas, we tended to see less biodiversity, but you see higher abundance often, just like we're seeing here, of one or a handful of organisms.
Benjamin is early today for his shift. He's accidentally slept through the 2 previous night ones. Party.
Alright.
Because he's been partying in the mess, so he's awake for his shift. The young, the youth these days.
Have you sampled the brachiopods?
Brachiopods? Yes, from the last. Yes, with Teresa, yeah. So I'm after these shells called brachiopods, which we sometimes see if we come across rocks. But the funny thing is, because we've been collecting them alive, I only want the dead shell. But there's a lot of biologists on board, so they come and quickly take the animal. Because I was just going to bin the living thing, chuck it back overboard. Like, I just want the shell for some chemistry analysis. However, the biology guys are definitely very interested in the animal inside, so sharing is caring. It's been very nice. I get my shell, they get their animal. Perfect for everyone. We found some, actually today, the ones from, I think it was the last just before we came up, in the really shallow bit. They sampled me a couple of brachiopods and they were so big, I actually had to go get extra large sampling bags out. Not extra large, but like larger than all the other ones I've been using for brachiopods. So, quite interesting, because then I was quite excited, thought maybe they're like really old, you know, like maybe 400 years old, and they look like a different species from that really shallow mountain bit as well. So, I'll need to take some good photos. Maybe Erin, I can use your photo cabinet and send them to my brachiopod colleague
Yeah.
for identification.
Yeah, sure.
So Professor Fish shall be coming into the room in about 10 minutes, and I'm sure he'll answer all your fish questions.
I guess some people saw a leopard seal the other day.
Really? Oh,
Yeah, that's what I
wow.
heard. There was a leopard seal, like, hanging out around was it around the umbilical? I don't remember what someone told me, but they saw one.
Anna, Bedhammond is doing the biologging now, so you can relax.
No, I can do it. Like last time. Having sea cucumber.
That is a lot of sea cucumbers.
It is, so many. Thing is, this transect kind of has us in a valley. I don't know at what point it goes up onto the ridge.
I don't know, because we
Yeah,
can't
that's
really
fine.
see past waypoint 4 on that map, can we? So
No.
Copy.
After that.
Red dot.
Okay. Which way is we dodging? We have to go south. Okay. Okay.
Oh, but that will take us up the ridge actually, Erin, so
Yeah,
that
this
might be
might
quite nice.
maybe. Alright, so yeah, we just got word that we have
Due
to.
east now.
Copy, we'll follow you.
We just.
And then once we're past the berg, we can go back to north and have full freedom again.
Copy.
Okay. Yeah, we're just having to redirect off our planned path a little bit because there's an
What's that
iceberg.
pink stuff again? Yeah, what is it?
Apparently, it was suggested that it's like marine mammal poo.
Who suggested that?
Really?
That's what was suggested. Sunny advised me that Matt
So Matt Lee said that.
said that. So
Interesting.
it's
I
possible.
wonder what kind of mammals. Do you mean like a whale poo?
A seal.
A seal poo? Oh, there's a lot of seals around this area actually.
Yeah.
I think it's like basically now you just go to the mess, to our restaurant eating area, and you basically just watch the seals go by on icebergs.
Mhm.
I'd like to think of them as seals on a bed. Sunbed,
Changing course
sunbathing.
to 125 degrees.
RV copy.
You can tell we're getting tired because we're quiet. The
Well,
energy
I
is.
was
I
searching
need
for
some.
the mammals.
I need some. My third dinner. Let me get this chocolate out. Maybe that'll help. You want some?
Yeah, there's some cookies up here.
I was searching for the pink stuff.
Making all the crinkling.
Well, I would make some comments on rocks, but I'm not seeing any. So
Yeah.
I did get to poke one of Teresa's sea cucumbers the other day. It was quite satisfying. It's got this funny kind of. It's a bit like jelly-like, but quite a stiff jelly. Like it's quite
Mhm.
interesting. You just give it a bit of
Oh
poke.
no!
No?
Looks like that Adelie penguins.
What?
The Adelie penguins make this pink
Penguin poo.
It's a pink poo from Adelie penguins.
Well, then maybe guys, when
And
you finish
whales
your
is-
shift, quickly run
and
upstairs and
whales
check
is
out.
caused by diet of krill. So great, Sunny. You were right.
I was
Okay.
right.
Yeah, maybe. They found some
Look at it.
high, apparently a diet high in krill results in pink poo
Yeah,
for
it's
penguins
and whales.
and whales.
Because of
Bit
the
like-
diet of krill.
Because of penguin and whales, so
and pinkish
it's
crustaceans.
it's probably more likely to be whale poo than it is to be seal poo then, because seals don't. I don't think the seals are eating things to make their poo pink, so it looks like whale poo.
Oh.
We have seen a number. I don't know. We have
So,
some
and we
whales.
sampled some whale poo.
Yes.
Maybe I should have logged it. some whale poo for the interested
A
parties.
notification.
It
Be
is
careful
very
of the
much.
sea spiders covered by whale poo.
The sea
Thank
spider.
you for waking us up for our shift. Revenge.
Dinner.
For dinner. For breakfast.
Okay, so how
I mean,
we
if-
can log it?
Andy just made a very valid point. It's kind of like when humans eat a lot of beets.
Hola, Pedro.
Hey, Pedro.
So now we will make an observation saying,
Changing course back to 090 due east.
hello.
Copy.
Okay.
I'm going to stay for a little bit, some rock collection.
Sounds good.
Alright, so we're changing course again as we move around this iceberg to make sure it's out of the way. So that we can continue along towards our planned route.
Oh,
The other team is starting to enter the room, so
you guys, we sampled
in the next
some,
few
whale poo
moments we'll be
accidentally.
switching out. We'll have another team come in for the next couple of hours.
Sample a sea spider from Patricia and accidentally came in with whale poo in the same sample. So she's going to enjoy that.
Okay, let's see.
Oh, it goes alright. We haven't done too much. We collected the rock at the Nagonin, and we did it in a skin bottle. We haven't even seen any corals yet. There was maybe one that was on the side, but it wasn't sure. So, from what I understand from the instructions, the holothurian collection are going to be shallower. So I don't think Trace was getting up. So hopefully that's accurate because we've been seeing them everywhere.
Okay.
But similar depths as before, so probably. And then, yeah.
So have you done this rock collection? Or is that
We haven't done this one yet.
Oh,
We're
we're
not
heading
quite
up
to
to
4,
four.
and
Yep.
we're going around an iceberg right now. So,
So
you'll
Tom, I'm
continue
going
up
to stay for some rock collection.
before-
No worries. that'd be
after.
helpful.
So we're pretty much on pace, maybe a little bit behind now because of the iceberg, but yeah, it's been sea cucumbers and apparently whale poo. We learned that the pink poo is because it's a high krill diet, so penguins and whales.
Pigment. Okay.
So learn something new every day.
Beautiful. Don't forget to see it near the end.
Yeah, we haven't seen too many fish, but we figure still you got the better eye for what to try to collect.
Teresa may
See,
be
well,
interested in that.
Yeah.
Yeah, because she's looking at the diet, if they're selective feeders. I imagine holothurian are quite
green
light.
rock we want this time.
Whale
Yeah,
or-
I think I saw it. But
Yeah.
You saw a green rock?
We sucked up a little bit with the picnic coating, but you
I'll
know,
keep my
she-
eyes wide open after that.
Small, like
Double
Seeing it some,
quadruple
so I guess
espresso.
yesterday's thing took us off of keep seeing it, but
I'm ready.
you're welcome to grab a sea cucumber with a bucket of push cord.
So yeah, because it says
Oh,
samples.
that's on the food.
There is no Niskin bottle with the push cord today.
Okay.
I will do like some stuff later. Yeah.
Using jars to do their water experiment.
Okay, cool.
Oh yeah,
Alright,
so
that-
yeah, so we don't need to do the skin before the push course
No
today.
problem.
yeah, I
But that's
remove
not
it.
on our shift anyway. So
Yeah, it's probably not your shift. Just
it's
pass
like at the end of tomorrow, like at the end of dive.
Awesome.
Nice
Thanks, y'all. Have a
Good night.
one.
Sleep well. Pedro, do you want to do talking? Shall I sit there? Okay.
Want to take a lead today?
No Pedro doesn't like it. He told me off earlier. And I invaded.
What do you have one?
Get ready. I believe Professor Fish has now joined. We had some fish questions in the chat.
Oh,
so
okay. I'll have a look through
They
it.
want to send them again. I told them you'll be here soon to answer all the fish questions.
All of them. I know. She gave me a promotion.
Skipper Captain Jessica took the wheel.
It's a Claude Captain, Skipper, Professor Jess. Yeah.
Oh yeah, sorry.
Okay.
Hello, everyone on the feed. We just had our science crew change, so we should be refreshed and ready to talk you through what we're seeing and what we're doing. I'm Tom Lindley. I am a fish person. I'm a fish curator at the National Museum of New Zealand, Te Papa Tongarewa. And with me in the room today,
I am Pedro Martinez. I'm a Spanish marine biology working in Germany at the Senckenberg Research Institute. And we just started our shift. Let's see what happens today.
I think at the moment the vessel is just dodging an iceberg, so we're following along. Usually the ROV dictates the course, and the vessel just keeps up with us. We let them know when we're moving, they let them know when we're staying put. But of course, when it comes to dodging icebergs, every now and then that has to flip. It's quite a complex operation anyway, coordinating two vehicles separated and connected by a tether. But with the icebergs floating around outside, adds an extra element. So once the bridge has completed this maneuver, we'll come back onto our waypoint and continue with the transect.
Apparently we're after green rocks today.
The pink wasn't enough.
No, we just got another pink one just now. So it says waypoint 4, we need rocks.
Okay, and we want a green one. Does a green one exist, or am I on a snipe hunt?
I'm not sure. We haven't seen one here yet, but it'd be cool if it did.
A lot of big anemones here at 700 and almost 50 meters. And also we see big patches of greenish material on the sediment. This means that there is a lot of productivity in the water column, and most of this is also raining down to the bottom in form of marine snow. Holothurian, sea pigs. Oh. This is the time I should wake up.
I thought my alarms were bad.
We saw a lot of sea pigs at the bottom. Small roundish sponges. There is some fish. And also a lot.
That was ink, I think.
Ah, okay, so maybe a squid skip.
Yeah. I thought it was a comb jelly at first, but no, I think that was a little puff of ink, so we've annoyed someone. I've seen a few glacial squid on the cameras.
Yeah, so far we've just had like mainly sediment like this. We've not really had that much rocks or that much fauna, so I think we saw a really nice big sea star.
Oh yes, I saw that.
It was massive. It was maybe like half a meter approximately, maybe a bit more. Almost the size of my gravity cores. But
There is
not quite.
it's quite a slope here. Here there are some traces, but in general there are not so many traces, not so many as in other places that we have been.
But the sediment looks really rich. It looks like there's plenty
Oh,
to
here
eat.
we have some outcrops.
I shall quickly log the rock.
Actually, we're not going to be able to stop.
No worries. We're just liking the change of pace.
Yeah, now we're flying.
Wow, this is steep.
Yeah. sponge. Sea pigs are also called sea cucumbers. The ones that are living on the sediment, as we have seen, they are rather called sea pigs.
Yeah, come back, Rich.
Morning. Yeah, I'm free to move now towards the north. Do you want to drop a new waypoint for me?
That's a
Roger, one second.
huge wall.
So you guys are fine with going to waypoint 5?
I think they did want 4. Do you know how strong the selection that was? Or was it just a nice spot for rock collection? If we can cut across to 4, just because I have a request for rock collection there.
I wasn't in the dive planning meeting, so.
Do we have a feeling that's where the berg is?
Basically, I treated that fine.
Okay. Yeah, I'll follow the instructions just to be sure. I don't want to miss something that was spectacular, but I have a feeling it's it's just at this point somewhere.
Huh? It just came from north, so everything we could just go back north. So we have to go. Either they come this way or they come this way. I'm just gonna put it there.
Here there is nice hard substrate. There is some colonization on the substrate, but not too many.
It's not as mature and rich as we were seeing on the other sides.
How's that waypoint, Rich?
Yeah, looks good. Heading in that direction. Point 4.
Roger.
Yeah, I think a lot of our waypoints seem to be the only ones, inside this little like valley thing. So it's just been kind of like fine sediment like this with loads of sea cucumbers. So Tom, are you hoping to catch some more fish today?
I will keep an eye out. I'm doing fairly well.
There's a shrimp on the rock and some serpulid worms with these calcareous tubes.
I'm not quite sure what these little Argentine-looking silverfish are. They're pretty nondescript from above. So we'll keep our eyes out for a fist-sized rock, about 10 centimeters. Bonus points if it's green.
So many sea cucumbers here.
Oh, one that loads of little dots, that's also acceptable. Spotty rock, please.
The spotty green rock,
Spotty
please.
green rock, quite specific today.
Coming right up. By the looks of this, you'll be lucky with any rock.
Well, we did find the kind of reddish pink one earlier. I don't think it was actually pink, it looked more like a manganese over grace. But we did get a nice sea spider from the dresseur, so she should
There
be
are
happy.
some sea urchins that they cover themselves with shells in order to hide. And here you see on the shadow that there is a lollipop sponge. These are penniagone looking sea pigs. And here a little bit roundish, the Scotoplanus looking sea pig. So there are two species here. These two species we have seen all over the places that we have been diving. So they are very common in this place in the Antarctica.
What's on the ROV is the altimeter?
Straight back.
Straight towards right. Okay.
So like right now, when you see like it's kind of flattened out a bit, but when we go up slope, you'll see that the altimeter is really high, but you'll see it come pretty close to you.
Yeah, because I'm reading not 1.7, and that
Yeah.
doesn't look 1.7.
Yeah, it's like a little bit far out here too. That's close for now. So we're going a little closer.
690 meters, we'll continue climbing.
You see the film over there? So we can have the way back to the vehicle. So there's one in the front, one in the back. You can see you're going a little bit up slope down.
And then there's this undercarriage one over here. You can see the skids from below.
So are all these species of the sea cucumber the same, or are they all different?
We see two species here at the moment. A little bit elongated is the peniagone looking one, and the rather roundish with these four filaments actually is the Scotoplanus looking. These are two very common genera. We think that these species belong to this genera, but we would need to look at them carefully and maybe take some genetic sample to confirm.
There are loads here, but we're not due to collect sea cucumbers just yet. But if the latest site is anything like this, we'll do okay.
How many species of sea cucumber is there?
In total, wow,
Yeah.
I don't know that.
By like hundreds, thousands?
I think less than 1,000.
Okay, by a lot.
Yeah, many are also undescribed. So they're
Oh yeah, if you spy a rock.
But these
let me
ones
know.
we're seeing here, are they like Antarctic endemic, or are they found all over?
Yeah, we think that they are just here in Antarctica.
Oh, cool. Finally, a
Oh.
rock I
Oh,
can
there's
lock.
Yeah.
rocks.
Now I need a hand-sized
Yes,
one.
good.
I think these are just some exposed bedrock sticking out.
I've not seen many loose pieces. That's a neat
No.
urchin over there.
I think there seems to be, because we seem to be in this valley, there seems to be a lot of sediment accumulation here, so it's probably just covered all of the... Maybe we should have done a gravity core right here.
One of the
dangerous.
starfish.
Oh yeah.
There might be a loose rock there.
Yeah, the one with the crinoid on.
Yeah.
That's an unusual shape if it is part of that bedrock.
Yes, I like this rock also.
There's one I tried, that one might be a bit big.
Oh, and there's one in the
smaller
background.
behind it.
Yeah?
Is it smaller or is it perspective? Are they about the same?
Perspective,
But you won't have to disturb a crinoid then. Although we probably will when we blast over it.
Is this an ideal spot for you guys?
Very
for
nice sea
sampling
star.
now?
Yeah, this is about at waypoint 4 we had a request in. I haven't seen a green one, but it is speckly, so maybe that'll count.
Well, you'll have to compete with Anna. She sent Professor Rock a picture of the previous one we took, saying, look at how good this rock is.
It was a fine rock.
You checked out the rock picture?
I did. Beautiful.
Excellent.
That's the Comparative out here.
Oh, gosh.
I think this is connected.
Yeah, that one over there looks a more horrible size.
Promising. It's one on the back there.
Yeah. That one there's a... You were right, it wasn't perspective, it is slightly smaller.
A little bit alien.
The sea stars are huge. Do you think that one's feeding? It seems to be hunched up a little bit. That usually means it's eating something.
Huge, actually. Yeah, that's pretty big.
That rock on the right is big.
Yeah, it's pretty big. It's gonna be
No, I don't think we need any massive rocks.
No.
Okay. Alright, we'll just carry on. Though that one's definitely big, but it's Easter.
Once we got up close.
Yeah, it looks weird.
Yeah.
There you go. Leave
There's-
this spot.
There's hope. Oh, there's a big rocky area in the background.
Sorry, guys.
As much as Professor Rock loves the big rock, I don't think we need to fill up the whole fridge with rocks.
You can get rock greedy. There's a point where it's too much rock.
I agree. So there's so many my rock collection cabinets can fit. The sediment looks very fine around here with loads of that organic stuff on top.
Yeah, they're well fed. I'm surprised it's not got more life. Everything we're seeing is kind of mobile and a little bit short-lived. Did you say this is an area that may have been unlocked relatively recently?
I think so, but not as recently as the sub-ice stuff we were diving at, in the previous station.
It's true.
Kind of
It's
fish
like
we have.
50 years. I think we were maybe mentioning on the previous dive. So this area, actually I'm not sure because we're a bit deeper right now, but the stuff that was shallower, that was certainly maybe like last 50 years or so would have been a pinning point for these ice shelves as they retreated.
Huge ponds here. We had also big sea star. And we see also some fish.
And there's a notothenoid down there to the left. There was a couple of eel pouts hovering around, those ones that curl themselves up. But there was something that looked a bit different.
What was the lifespan of the sea cucumbers again?
It's not known for sure, but something around the decade mark.
Really?
I think it's quite surprising. I think Teresa found that one the other day. She was really happy there was like loads of caviar inside of it. I suggested she makes like a caviar sandwich, but she was not so keen. Actually, Tom, when we went to sort out the brachiopods this evening with Teresa, your fish, the whatever was in there, was very smelly in the fridge room.
In the fridge room.
Yeah, it was like this distinct fish stench in there.
It was gone though, right? I put it in the freezer.
Talk about this.
Oh, sea pen.
You can look at it? Sorry.
Yeah.
Oh no, we can. Oh, did you want to get it?
No.
No.
No, I was trying to comment that there have been some places where they were more common, these sea pens. It's the first one that we see here. Are we supposed to collect some holothurians?
Not at this site. I think she wants them to go shallower.
Okay.
So it may be towards the end of our shift, or it might not be on a shift at all. What we are hunting for is a rock. And just you wait, when we get to the Holothurian station, it's going to be full of rocks, green ones,
I
and we
think
won't
the
find any.
Holothurian station
Is
is
that
like
up
a
there?
then dive.
Is that a rock?
Oh, yeah.
Look how lonely it is.
Oh,
We'll give it a home.
Can join all its friends in my rock
You
collection.
want to collect it?
Yeah,
I do.
that looks the perfect rock. That's Manip claw size.
It's good size.
Not too much rock, but enough rock.
We're just gonna collect this.
okay, we're landing to collect
See if
this
something else to look for.
drop stone.
Can we go slow forward?
Here we see
See what the current's doing. Looks pretty slack. Yeah, the perfect current on the last one. It was just coming towards us just enough to clear sediment quickly, but without causing issue. No, this is pretty good. If we could lift it up in front of the camera and just have a look at it, take some stills,
Yeah.
and then we'll be able to tell them apart in the box. Nice.
oh, there's a Brius one on top.
This is 790 dive, Pedro, yes? Tom?
We get that high? Yes, 790.
Thanks.
Great, thank you. And that can go in the rock basket. Maybe the outboard bomb, rock basket 1.
Outboard. Rock basket outboard. Yeah.
These dives can easily go for 14 hours with multiple shift changes, and we have to keep tabs on where we put everything and where it came from so that our records match up. So a lot of things we have to put in separate containers. It's a bit like, you know, that little thought experiment where you've got to get the grain, the fox and the chicken across the river. Remember that one? It's kind of like that. There's certain things we can't put together. There's certain things that will escape. So some of the bio boxes are divided into 4. But if you put something that swims in there, you kind of lose access to all of them. So rock in rock basket 1.
Thank you for the rock, guys.
No worries.
Okay, it's gone past my third dinner.
You need more rocks later on?
There's a couple marks on the transects. Yep.
Okay, well,
So I'll just follow the rules.
yeah, it's gone past my third dinner o'clock, so I shall see you all later.
See you.
See you later. Enjoy dinner.
Thanks.
So yeah, we have to keep good logs of where we store everything. We have to keep in our head what's in where and what might interact. Some things slime, some things eat each other, and some things are known escape artists. We've got quite the kit on board for collecting different things. On the far left we've got some coral quivers, which are like tall, upright containers. They're good for long, thin things. Behind those we've got a rack of Niskin bottles mounted on the side of the vehicle. They are open tubes that allow water to flow through, and then if we're at a target site somewhere we're interested in the water, then we can close those and that traps the water from that area. So that could be an oceanography thing, looking at chemistry, or it could be eDNA, or there's even a microplastic study going on this trip. Then in the center we've got these huge bio boxes. big boxes with hydraulic lids on them. Some of them are subdivided, so that allows us to separate the animals. But like I say, if they're swimming animals, then once you put something that's a flight risk in there, you have to keep the lid closed. So we coordinate things like rocks and sessile organisms, things that won't move, and then you put a mobile one in last thing and then leave that door closed. In front of that, we've got all the push cores, which are a sediment sampling cores. So you lift them up with a manipulator arm, push them into the sediment. Classically, we would do things like that with a multi or a megacorer, something we lower from the ship, but we're kind of going off map resolution then and firing blind. They quite often hit a little bit of rock and shatter all their tubes. The ROV gives a great opportunity where we can be really selective about where we put those cores, and also we've got then the images when you come back to analyze, well, why was that core different? You can see the sort of area it was taken from.
There is a big crinoid with the arms extended at the bottom. We see a lot of shells also, these white rounded structures. These are shells from dead mollusks, but of course there are some living ones in the sediment. We cannot see them. And there is a brittle star, sea pigs, and here there is some
Right,
jelly,
you can continue with move.
jellyfish in the water.
That's beautiful.
Yeah,
I understood, getting underway.
that's a 1004 and 2 shrimps. Very beautiful shrimps.
Ah, bipod fish.
Here we go.
We've just started calling them bipod fish on this one. That's one of the chinnichthids. They're two main groups. Some have long stilt-like pelvic fins and some have little tufty ones. I think that's the one that you gave me from the vehicle the other day. Oh, bow wave catching up. Oh, look at it walking on the stilts.
Oh, really? Wow.
Let's try it.
Very nice shot.
Oh, that's great. Yeah, it looks like... Can they fold them out?
You know, sometimes when you run with your arms behind your back.
Yeah, doing the Naruto run.
Oh, now we started to see a lot of small blackish pebbles or gravels at the bottom. We didn't have this before. These are very small little pieces of rock. And yeah, there's during last dive we had a lot of krill.
We had a
We
fan
will-
club.
Yeah, we will have probably this dive also a lot of krill when we get a little bit higher. We had them about 300 to 250 meters.
Were you interested in taking a sac sample at the gravelly site?
Yeah, this would be a good idea.
Yeah, do you want to come down and do it? Shall we grab one here?
Yes.
Yep. If we come to a gentle stop, we'll take one of the Sakitos here.
And it looks like Eco's already been taken.
Doesn't look like it, but I felt like Aaron mentioned I might have. We've got no Sakitos taken.
No,
No.
I don't see that.
No,
Yes.
I think we're good. Any of them?
Any of those Eco's really.
Yeah.
And a gravel pit, basically. The idea.
Yeah, I
Yes.
think it's fairly consistent here. And we got a scallop again. That's the first one I've seen, I think.
Yep. There's a shell of a scallop.
So I'm gonna try by heading a little bit more.
I'm not sure what these silver, sort of Argentine-looking fish are, like the one in front.
You feel that they're different from?
Yeah, it's not those silvery ones that have been bothering us because it doesn't have the beak. The other ones were barracudinas, but that's something else. I don't know who folds these backs. It's like a turn-down surface. They're immaculately folded.
Okay, with this bag we can collect a big amount of sediment from the surface. I guess that we will have there 3 to 4 liters of sediment. This is much more quantity that we can get with the push corals. And then we will process this sediment sample, sieve it and look for little worms and little mollusks and shells and gravels that are in the first centimeters of the sediment.
Because the cores are about sort of sediment structure, aren't they? They're about the layering. But as biologists, we know all the good stuff is in the first couple of centimeters, so this is just a way of getting huge quantity of the good stuff.
This is then a qualitative sample, so we cannot estimate densities of organisms with that because we don't have an exact reference of how much sediment we have collected. But for the quantitative estimation of the density of the animals, we use the push corals.
Coming to a pause.
Do you think that's full enough? It's looking pretty full to me. Or is it?
Underwater fan.
Is that bag one?
Is the, yes, bag one.
Little stack.
Very still current. You see it change quite a bit even over the dives though. The topography is quite complicated, so you end up with sheltered areas that are quite low on current, and then a few of them we've rounded the top of an outcrop and suddenly see a little bit of elevated current, and then you get all the filter feeders to let the current do the work for you and just hold a filter into the flow.
I was doing it blind. It's amazing. You guys can see it in the look down here.
We don't see anything at the moment. We need to wait a little bit.
Oh, sorry, we're looking at a different camera.
And get a slightly better view down from above. Not by much.
You think it's refull? Do you want me to pop up?
Yeah,
the
look-
or do we want it to be from the same side?
It looked like you got a fair amount.
I'm gonna come up.
We'll periscope up and see what's going on.
Unlike it.
It's fun.
Nicely done.
It's looking like a full psyche book.
That's nice and full.
Fish come in.
That's the Barragadino ones. Those are the ones that have been bothering us, but those silver ones back there are a bit different. Okay, Sakito number 1. 734. I think we're good for rocks for this section. We can head on to waypoint 5, and then we've got a video transect where we have to try not to get distracted by exciting things, but I bet it'll happen. We're only human.
It's almost impossible.
It is. Why does that one look different?
The skull goes
Would that,
itself squared up and a little different.
would that not be very big if it is? But that's what my brain was saying as well. One of the Dumbos looking all melted, but that would be a big
That is
one.
melted temple.
And I think it is.
Oh yeah, that's a cephalopod.
So that's pretty big. That's a mantle length of what, maybe 20-25 centimeters.
Sleeping.
That is a nice color. That's a pretty big and old looking one to me.
Yeah. Very nice one. We'll let him sleep.
Yeah. We're not interested in collecting them, but it's very cool to see them.
Honestly, there's a seafloor of those.
Yes, this was kind of an octopus. While the genus Octopus, I think, is not present here in Antarctica, this belongs rather to the genus Elidone or Paraelidone. These octopods, they have just one row of sucking disc in the arms, and the octopods, they have 2 rows. But yes, it was an octopod.
Wait, you could resume move.
Here we see again a lot of clams.
Confirmation from
Was it
chat.
the-
Yeah, it was a big Syrioctopus. Syrioctopus. Looking gravelly again. Can we zoom in on that fish? Do we have time, or are we going to wipe it out with a bow wave? Don't worry, I was just curious.
No, no, we can try. You can always try.
I think that's a nice fish. I don't think there's 2 dorsals there.
Holoturians swimming. Oh, don't worry.
Nice. Oh.
Yeah.
Could have been a Bathydraco, but I couldn't see a second dorsal fin. Oh, there's another one. It looked like a glitch in the Matrix. It's exactly the same size, same type of fish.
Probably cows. We're going to talk about this afterwards.
Yeah, confirm what they saw. I know what I saw. It's a thing in the sky.
See that alien up there?
Yeah. I tried though. I thought about it. I thought very hard.
Going up the smart rocks here.
I think we're good for rocks for a while until waypoint 7. Oh, nice ice. Sorry, it's a notothenoid. That is nice.
Oh, now we have a lot of rocks here.
It's too big, but I can enjoy looking at it. So if we do see a pink or a green one, we'll get it, but it's not a site we're requested to sample specifically.
Yeah, I think that the suggestion in the chat is right about the genus Cirroctopus. I have been checking that. Oh, there's another one, another octopod here to the left. Seems that there are not so many records of this, but they are living in the Antarctica.
Yeah, I recognize the names in the chat, and you can believe those. And so you start to recognize the community that watches along. And
You're in South Africa, decided to just come on right away.
now that we can share things like this live, it's lovely to share it anyway, but it's amazing how knowledgeable the community has become and how structured it's become, and it's really benefiting science. There are multiple citizen scientists I can think of who've either become experts in their own right and provide really, really useful feedback, or become sort of networkers and are good enough at IDing a wide variety of animals, but then know the experts and know what platforms to contact them on. And so much science is getting done just by the community that have built up around these live streams.
Also, it's only recently that we have in situ photographs and video of many of these species. In the past, they were collected with trolls, and normally we have only some collection photographs. Sometimes the animals, they look quite different when they have been fixed in ethanol or in formalin.
It's crinoid. Is that some of the whale poop in front of it? The red?
Is it whale or is it seal poop?
I don't know.
Red balls.
We've been seeing some little patches of red sediment, and the consensus is it might be marine mammal poop. Things that have eaten a lot of krill. I get that roller coaster feeling. Just coming over the rise.
Small. Huh. I expect those rocks. A lot of rocks.
I'm not seeing anything in the way of corals or any mature growth on these rocks.
That's
I've seen a couple of sponges, but not the huge ones we were getting in the others. Not the sort of long, slow-growing things.
that's true. Well, this time of the year in Antarctica is summer, and we are below the Antarctic circle, the polar circle. So the sun is not getting completely down. So we have also you can see the midnight sun. But of course, the light is not coming down all the way down to the bottom. We are now at 650 meters. So the sunlight at these waters that are so productive probably only gets a few hundred of meters. Some shrimp in the water. We are continuing to climb up through this little canyon.
I'll start with like 5. You guys want to continue to 6 or?
Yeah, once we get to 5, we'll I've asked for a transect along there, but I feel like we've gotten some good transect in already, so if something really leaps out at us, we'll think about collecting.
Roger.
But yeah, we can enter transect mode. Less than a meter altitude if it's safe to do so, and about between 0.2 and 0.4 for speed. What's that up there? Oh, it's another one of the ice fish.
So they cannot retract these fins.
It doesn't look that way. I thought they would have swept them along the body. That's incredible. The way you can look around, picking where to land.
Yeah.
We've been given these the nickname bipod fish. This is the Chinichthids. This is the crocodile ice fish, or the true ice fish. So these are the ones with the white blood full of antifreeze. And there's 2 main groups. Some have little rounded pelvic fins, and the others have these huge stilt ones.
Here you go, landing. Oh, oh,
Oh.
oh, oh. This was a hard landing.
Not so graceful.
Interesting picture.
Looks either well fed or.
There's a lot of them.
Oh, yeah. So you think that's the other type with the that lays flat?
You want me to get a better shot of them?
No, we can move on. We can move on. That was a lovely shot, though. It's great to see them swimming. I didn't know they swam that way.
So we are in an area with much sediment. We have a little slope, some crinoids at the bottom, some anemones, and of course our holothurians, sea pigs everywhere. They're very, very common here. They are grazing on these little patches of freshly deposited material. And also some fish.
I think that's the other type. So that's the same family as what we've been nicknaming the bipod fish, but with the little round pelvic fins rather than the long stilt ones.
We're pretty much at root 5 around here.
Grand. Guess we just continue as we're going.
Roger.
If we see cool stuff, we'll look at cool stuff. If it's really cool stuff, it goes in a bag.
It's really cool stuff with those.
Providing an evolutionary pressure to be interesting but not too interesting.
Let me know if you want to see anything. There's also future vision here.
There's a little fish on the way up. Nice and pale. I think it's one of the notothenoids. They're usually a silvery colour, but that's really pale. It's got a friend on the rock. There's a very good chance that they can adjust their colour.
Oh,
Oh,
you
cool.
failed to see that.
So one of them's being a rock and one of them's being sand. They are different species, but still. They were nicely arranged by colour. We don't have any sunlight making it down here, but there will be bioluminescence. It probably makes sense to match at least the contrast of your environment. Nice, and that distinctive spot on the first dorsal fin, I do recognise that one. Terranovi, is that something?
And the holothurians have sensed that we are in the vicinity and they start flying, swimming.
All good to proceed.
Yeah, all good. Just enjoying some browsing.
There's a sea pig who's tentacle like blue line. So I'm just gonna-
Because the vehicle is longer than it is wide
You were
on
quite
some of
right.
these slopes, we can sidestep our way down. Lets us get a little bit closer to the seabed.
Updated waypoint.
Strafing. No, no worries. Fairly steep slope we're coming down due to the orientation of the vehicle. It's difficult to go downhill. Well, it's difficult to get the cameras close to the seabed going downhill. Another octopus on the far left. Field upon field of
Yes.
sea cucumbers. These fish look very flighty. I don't think we'd be grabbing any of those.
Crinoid and a big brittle star. So they are not so common in this area. We have been many of them in other places. And probably when we get a little bit shallower, they will get more common.
The sediment is looking cleaner here, so they're doing a good job cleaning all this up.
Yes.
Because it.
Leads of the sea.
What's that?
Said Mary leads of the sea.
Because the conditions can be so tricky down here, we often see, and actually just the ice and being able to access this part of the seabed, we often see these areas in the summer, so just after the spring bloom. So we see them when there's been lots of input into the seabed, there's lots of krill, the sea cucumbers are all well fed and breeding, quite a few of them have eggs. So we get a glimpse of the feast basically, and we often miss the famine part. So we think the spring bloom, judging by the oceanography measurements, was just as we left port. So all this marine snow and energetic input to this community is kind of at its peak. This is as good as it gets, and then it'll start to run out again. Everything will reproduce and then go into a sort of standby mode, waiting for the autumn bloom. A sort of ability to collect data is one of the things we have to really watch out for in science. It's very easy to draw the wrong conclusions just because of where you can get data from, basically. Like loads of studies on people, especially survey-based studies, tend to be done on students because students are quite willing to fill those things out. So then you end up with sort of bias towards that slice of the community. And we see the same in deep sea stuff, like we could only really get sediment samples before we could be as picky as we are with the ROV. We could only really get sediment samples from big sandy areas where we knew we could drop our corers down, and so that was all we had data for. We had to extrapolate the rest, and it's a similar here where we can only really access these areas in the summer, and then we have to infer what's going on in between. So that's why long-term observatories are so useful. Give us that sort of fill-in long-term data, make sure that we're not measuring some dots along a oscillating pattern and then drawing a straight line along them when actually it's cyclic. Interesting jelly coming up.
Yes, nice one. Oh, it is coming out of this hole.
We can keep going. It's a nice one though.
Yeah, but you see this hole?
Yeah, something darted in, didn't it? And
Yeah.
kicked up some current.
Now we see a sea anemone. And also there are some acorn worms in therapnoist. These are producing this zigzagging pattern. Maybe we find a good one to take a shot.
I feel like the diversity has taken a little jump. It's subtle, but there's not just sea cucumbers. So the conditions allow us to adjust the position a little bit so the lasers are a bit
More on the
closer.
front?
Yeah, and if we can get lower.
Change my heading into the slip, and we can definitely do that.
Oh, that's great.
I'll just let it roll on.
So on these transects, what we're doing is we're using these two little green pixels you can see in the center of the screen. That's our laser scale bar, that's 10 centimeters apart. And we try and fly at a fairly consistent speed, about 1 meter off the seabed. And this allows us to get quantitative assessment of the animals. So it's all fun and games when we're exploring, but we're gonna have to watch all these videos and count all these animals. And then we can figure out animal density and even biomass. So how much animal material is down here, how much energy does it take to feed a community of that size. Then we can back calculate how much energy is entering the system. All fun things like that, because we love to draw graphs and there's sometimes maths. And there's one of the acorn worms in its slime house. on the previous dive, there was some weird sort of impact craters in the sediment, and they didn't always make sense where they were. We thought maybe they were iceberg drop stones, so as the ice melts, it drops the stones and things that it's picked up when it rolled off the land. But we couldn't find a rock in the bottom of them, and some of them looked like they sort of scraped uphill. It wasn't the icefish nests, which, if you haven't heard of that, certainly look that up. That was discovered a couple of years back, that the icefish make these elaborate nests and all gather in these nesting grounds. So it wasn't their little bowl-shaped nests. It looked more immediate and destructive material had been sort of thrown out and thrown to the either side. And I'm thinking, based on the depths as well, that it might be diving marine mammals snuffling around in the seabed. So you get things like beaked whales, and they have an advantage because they can use their echolocation to actually look through the sediment and see if there's tasty things buried there. I think there's another octopus up in the top. But I think the seals do it as well with their whiskers. I think they root around in the sediment. So we think that's what we were seeing.
Although they were quite deep. I mean, okay, we were 250 meters, and this is probably the limit of some of the seals.
Elephant seals, I think, have topped 1,000 meters.
Yeah.
And the beaked whales can easily forage on the abyssal plains.
Yeah, the words they can, yes.
Some record breakers.
Another wonderful sea star.
It's only a tiddler compared to some of the ones we've seen.
Yeah. And also very big crinoid here.
Mm. We were talking about their swimming behavior before, and about the sort of distributed nervous system on sea stars. They're kind of democratic, and I think each leg individually decides to swim, and then it makes a consensus. Because a few times we come past and a few legs are sort of paddling like they're swimming, and the others haven't really noticed. And I don't think they've got the brain power to coordinate their swimming. I think each leg just decides to swim, and when enough of them decide, they pull the legs holding the ground off. There's not much decision-making power in them.
Want me to deviate from transect and kind of inspect this out?
Yeah, we might as well. It looks like a different habitat. Strange, is that just where the sediment slipped or something? Yeah, it's just where it's caked on and then slipped away. We're surprised at some of the angles that these sediment is holding onto. It's really quite cohesive and cakey.
Yeah, we see some shrimps. But yeah, they're not so populated, these walls, in comparison with other places we have visited here.
Yeah. I was still on the lookout for corals, but I'm not seeing anything slow growing. I think this is going to be an interesting station when it comes to comparing.
Go look in the rock and then I'll go ahead and get it. Focus on not doing us any favors here.
These slopes require a little bit of finessing. So there's a vehicle roughly the size of a minivan with a tether coming out the back that you've got to have sort of held in your brain at all times. It's easy to sort of fixate on the camera and become the camera and forget how much there is behind you, creating bow waves and thrusting off sediment and mixing it all up. And I'm just glad it's not my job. I just get to watch the screen and make unreasonable demands.
Yeah, there's a fish following us.
That's one of the barracudinas, Antarctic Jonas fish. Keeping pace, they show no fear with the ROV, and despite never asking for them, I think I've got 5 of them in the collection now because they keep wedging themselves in the ROV.
We are sliding down into a small valley. The waypoints that we have for navigating, we are going to visit some hills, but of course between the hills we need also to go to some little valleys.
There's one fish that's just like, you see it here? So they just ladder them with this.
We've got our first fan.
It's been really nice for me, but
On the last dive, if we stayed still anywhere, we'd gradually gather more and more krill. They just really like the lights.
Here we start seeing some sponges also, these round sponges.
Okay, getting a bit of a habitat change. I think the whole ecosystem here is propped up by krill being a little bit daft, not very bright. We have a low light mid-water camera as well, and we've caught some nice bits of bioluminescence. So we've got what we're pretty sure is a krill spitting out a flash of light like fireworks. So they throw light out to sort of try and confuse predators. It's not going to put off a whale, but something equally krill-sized might be put off. And then on a more recent dive, we got what we call a pseudomorph. So some animals use like a smoke screen. They throw out bioluminescence to sort of confuse and dazzle their predators. Just a side note, that sort of red staining on the seabed there, that's what we think is marine mammal poop. Remains to be decided, but we've seen it at a few sites, but I'm seeing it more here.
That's so, yeah. Maybe from the crab eaters.
Yeah. But yeah, another way of escaping with bioluminescence is to create a pseudomorph. So some animals bind up that bioluminescence in mucus, and so essentially you create another you and disappear into the darkness and leave this glowing shape behind. So it's bound up, it doesn't just diffuse like regular bioluminescence, it keeps a shape, and so a predator will then go after that. So it's like taking your shadow off and throwing it at someone to distract them. Oh, and good input from chat as well. Yeah, and sometimes that sticky, glowing mucus sticks to the predator and just paints a target on them, essentially. It's very cool to think about what the environment is like for these animals, because of course we're throwing artificial light on everything, and that's how our eyes like to work, and that's how we like to navigate and understand things. But these animals are usually producing their own light and observing their own light. And so that makes the equation much more open to manipulation. You know, when you control the light source and your appearance, big things can appear small, small things can appear big, you can pretend to be things that you aren't. And basically it's all about remaining undetected. You only have to sidestep into the darkness and you can disappear, but even the water is full of bioluminescent bacteria, so even moving too quickly will set off these burglar alarms and reveal your position. And there's some lovely footage that somebody shared with me recently that these sea pigs twinkle and sparkle with bioluminescence when they're annoyed. Some great low light footage of them sort of looking like a firework display. So these animals never see themselves the way that we see them. They see each other by their own and each other's light, and that allows all sorts of different types of communication. So when we shine lights on them and look at them like this, it's essentially like an X-ray. You know, they're never seen in this way. It's good to try and get your head around that when you're trying to work out what these predator-prey interactions are like and what this is like as a habitat.
So we just passed waypoint.
We just keep soldiering on. I've got a note here: evaluation of ice conditions on the eastern side of the transect. I don't know what that means. Should we evaluate, or is that they think there might be ice for the bridge?
Yeah, we can check the bridge. Is it gonna go east? Is that? Well, we're going east for.
Gets a bit zigzaggy. But I think the bridge would have let us know if there's anything to worry about.
Yeah. Just carry on.
Yeah.
Until it's not too.
If we hit ice, we'll know.
Updated waypoint.
Copy that. Updated waypoint.
Is that a big sea urchin right there?
Yes. And what is this?
So here's something interesting.
Scarf.
Weird little scars here. You can see
Yeah.
it continue up once as it can.
Something has scoured here for a long way.
Yeah.
And is this coming away from the ice shelf? I'm trying to orientate myself with the surface.
Look at this. So we're heading 150 degrees.
Yeah, our heading is 115.
Okay. Don't remember where we are to the shelf.
Kind of just stops there.
It does. And does the elevation change much if it was ice touching down? Or is that some large animal that's face planted and explored along?
It was very straight line.
It was. Another octopus ahead.
Yeah, here we go again.
I don't feel like the elevation has changed much. I feel like if something had made contact with the bottom that was floating along, it wouldn't have left when it did. We'll show that to the geologists, see what they think. ticked off many of our wish lists yet.
No.
Not seen any coral. Very occasional sponge. We've got a pink rock, but I'm sure they'd like another. I think they're making up green rocks. Lots of sea cucumbers and lots of little coiled up sea cucumber poops. Detritivores are weird in that their poop is probably cleaner than what they eat.
Yeah, there is also an ascidian sea squirt.
Oh yeah, one of the predatory ones. It's alright, it woke me up. It's better than
Sorry.
coffee. Oh, I keep forgetting there's not a back to this desk. I can straighten my legs. Probably don't come this far down, but there are commercial fisheries in the Antarctic Ocean. They tend to be long lines, and they tend to target the toothfish. There are a couple of other target species, but the toothfish is the main one.
There is no bottom trawling, is it?
I don't know. No, I don't think so. I'm not sure it's allowed, but there is long lining. A lot of we've got quite an extensive Antarctic collection at the museum in New Zealand, because we've got a good relationship with the fishing community, and New Zealand has the Ross Sea dependency, so we're lucky enough to get a lot of samples donated by the longline fishery and by the fisheries observers. It's a really useful input of data, and so part of the reason I was curious to come on this trip is to see what it's like on the other side, basically, so on the other side of the Pacific. Some similar species and some endemics only from this area, and those are really important for comparison, basically. So if our collection can have the sort of sister species from either side, it's a really good comparative material.
We have not seen any fishing vessel here. In the Bellinghausen Sea, we have not seen any other vessel. The only vessel we have seen is the Sir David Attenborough icebreaker from UK that was performing some activities in a nearby. But there is some vessels. There is some tourist vessels. If we look at the IES signal that some of the vessels have, we see that there is some tourist vessels, also some laser boats, but we didn't see any fisheries vessel.
this site's going to become a good comparison to the other ones we've been looking at. Things feel quite different here. Really sea cucumber dominated, but nothing long-lived, slow-growing, even on the rocky outcrops. They're all things that can colonize and bounce back quite quickly. One of the big advantages of this current cruise is we're massively multidisciplinary. We've got oceanographers who are helping us decode the chemistry and makeup of the water masses and how they're likely moving and how they're likely changing, especially when we're not here. Deployed some gliders to do that give really high resolution results, and you can just set them free and tell them what to do. Actually, the pilots are back in the UK. It's amazing, you can throw something in the water here and it's being piloted back from the UK. And then we've got extensive geology teams and mapping teams, both rocks and sediment specialists. And it's unusual, you'll have people with a little bit of interest, but never so broad. And everything bleeds into everything else. Everything is connected. So it's been really, really useful to have these different communities on the vessel. And, we'll be scratching our heads about why something is a certain way, and then the geologists will chime in and it's like, oh, well, it's only been like this for 20 years, and then it all makes sense. It's been really useful to have this community. And then now we're in the digital age, that extends well beyond the vessel, because I'm seeing the chat scroll by with things I didn't know and making connections I didn't know about, and remembering dives I was not on shift for or asleep for. And it's drawing all these connections together. So the pace of science has never been so fast, and it's never been so inclusive. It's nice. Something on a stalk.
What is that?
Can we go look at it?
We might as well have a look.
Alrighty, let's do it.
Oh, something different. One of the sea pens. There's not much of a crown on it.
No.
Is it retracted?
Maybe that. Ah, it's retracted.
It's sleeping. We'll sneak past. I didn't realise that crowns could retract quite that far. I like how the radial ones all retract downwards, and then that poor one in the middle has no choice but to stick up. It's a sea pen, apparently.
Oh, okay.
And then there's a little swimming polychaete. Looks like it's going to collide with it. Well, that was a big curled up eel palp there.
Yep.
Just there.
Can I look at it?
Yeah. Do we want to mix things up? Do we want to go fishing?
You always go fishing. You want that as a sample?
Yeah. I
Wait.
reckon. Is that okay?
Can't try to collect that one. You want to get the fish?
Yeah. Yeah, I'm tempted to. They're not as quick as the other ones we've been seeing. The slurp gun might be best, if you approach from the head.
You're getting them first before you
Before I go disturb it?
see them.
Okay. Let's take a quick shot of it then.
Nice.
Shot there for you, so
Yeah.
that's what it is.
They're incredibly tricky to ID. Even when it's in your hands, it's difficult. They're a really interesting group. So they originated probably in the polar north, found their way down here and then just exploded into loads of different species.
I'm gonna back up a little bit and then get scared.
So they had an Antarctic radiation.
Yes,
Oh, yeah, that's great.
and then that may have seeded the deep sea. We're not quite sure. They've got a complicated history. There's a few studies that are trying to use their genetics and genetic clocks to try and figure out just how.
John, we're gonna try the
Yep.
net first.
Oh yeah, sure. Dealer's choice.
You know, if you want me to hire someone to do it for you. Nice.
I'm reaching in our golf bag for the appropriate
Wow.
club.
It's the first time I see this in action.
Alright, we're looking for approach.
Where'd we leave that fish?
He is right here.
Uh,
He's right there.
Keep the eye on the prize.
Just didn't want him to get scared while we're doing all this enabling.
A good call.
I'm down for a sec.
Looking a bit wary. But hopefully the vehicle's the big distraction.
I was thinking what to do.
Well, the other ones were super easy to catch because they curl up. It's a terrible predator defense. Nice. Oh. Ah!
Escaped.
That was the fold in the net. That was a brilliant aim.
You got it. French, you can come tell small stuff.
Copy that, stopping the show.
They're not great swimmers. They don't have a huge range, but it may have bolted.
Happened to me yesterday. Oh, sure.
Yeah.
We see another one, kind of in the vicinity.
That livened us all up. That was some good fun. Aim and technique was spot on, but the net just tangled.
Let's go for a fish hunter now. If we would have signed backwards, we would have seen kind of like dust. Although now it looks like there's no more fish.
That was the first one of that type I'd seen. There's some other ones from the family, the little ones that donut up, but that was the first big chunky one.
So you want one with a donut fish.
starting to level out. So according to the map, we're sort of hitting the bottom of this little ravine before we follow it along and head up. But I think based on what we've seen so far, the ravine itself is very full of sediment. We've only seen a few spots with some exposed rock. So not many chances for sessile things to find a foothold. A few more sponges here.
You see a large nemertini here at the bottom. This white, long worm.
Are you interested in said like white longer?
Just passing comment.
Roger.
There's a fish back there hiding under a sea cucumber. Lots of detritus down here. So all this marine snow we can see in the water column, that's raining down from the surface waters. We've probably had the spring bloom a month or so ago, and lots of dead phytoplankton and poop and dead things and molted skeletons are all raining down, and that's what feeds this community. And you can quite clearly see on the seabed the darker, greener, enriched biological material. And that's what all these sea pigs and grazing detritivores are feeding on, and they're revealing that paler, cleaner sand. So that's what's propping up this whole community. There's two major groups. We've got the deposit feeders, so the ones that are snuffling up things once they land on the bottom, things like the sea pigs and the sea cucumbers in general, and then we've got the filter feeders, things like these sponges or the corals on some sites. We were saying earlier that this is the sort of feast stage of this community's life cycle, so this is when everything's gorging itself, growing as quickly as it can, producing gametes and reproducing and releasing as many eggs as they can before things get a little bit more meager. So even though it's warm and sunny and summer up top, this community is limited by nutrients in the surface, so it's usually iron limited. So that's what halts the plant life from growing and propping up this whole community. So there's a explosion when things start to warm and they get more sunlight until all of the resources are used up, and then the growth sort of halts until it comes around again. Is this floating in the water? It's one of those guys.
Yeah. Oh, now we see it. Ornithurium
Yeah.
poopy.
Getting a good spook. So that's one of the eel pouts.
We have seen a number of different sponges here.
Another one of those predatory tunicates, predatory sea squirts.
That one over there.
These behind your eyes are probably more intuitive, right? That little guy?
Oh no, it's
Just saw a coil.
saw a coil. Oh,
Although there's the mysterious red poop.
saw a coil and
look like these lollipop sponges. But they may also be a sea squirt.
It's a large sponge looming in the distance. Ah, do you spy? I think that's a notothenoid. Yeah, I think so. That's probably going to be quick. We can give it a try, but I think it's going to be quick. I think it might be a fruitless endeavor.
Set. One set. So if it takes off, you can chase behind it.
That's right.
So like coming up straight up. Well, I'm gonna put this in front of you. Put the arm in front of you.
Roger.
You just kind of glide in, and I'll just assist whatever.
Great. I like it.
Yeah, this.
ROV bridge.
From my bridge.
Yeah, ROV. I've just got to move a little bit further south just to avoid a small growler. So it shouldn't take us off track too much.
Yeah, no problem.
I think let him do the maneuver because this is. I knew that would be flighty.
Alright. Roger. You don't want that one anymore?
I think we could be chasing it for a while before we exhaust it.
Just
Oh, yeah. Let's try that.
Okay,
Let's do
jump on it.
Claws on it. Dun, dun. Dun. Oh. That fish is going crazy. And also,
yeah, no switch.
At less than 1.5 degrees, they've got that kind of movement.
I think he's right there. But
They're very wary.
I'm not sure if he is.
he's still right there. It's okay. Okay. Roger. Alright.
Yeah.
Lives to fight another day.
Saved by an iceberg. The fish has no idea. Might not even know what an iceberg is.
Yeah.
Can you just go?
Alright.
Oh, it's become a quest now. Claude's got quite into the fishing.
Long one.
Alright. I've never fished in the sun before.
Do we need a scoreboard? Do I need to give a points value to each fish based on its flightiness? And we start a roster.
There's one up there.
Yeah, is there any
He's
fish
logged
out
on.
here that you like?
Come down and just. With silent mode, just.
These are the same ones, right? These are the ones over here to the left.
Yeah, they're a little different. I could admit I'm not sure what these Argentini-looking ones are. I couldn't give you a score for how quick it's likely, but that looks pretty quick.
Do you want any of these? There's also one more up there by the sponge.
Yeah, there's a notothenoid up back there. I think they're both going to be pretty quick. I mean, have a shot at both and see how we do.
Can I shot at both? Anyone that's preference?
Oh, you want this?
I think you won this.
Alright, do you want one? That one?
Yeah, give it a shot. Give it a
Okay.
shot. If we don't win, we'll move on. But I think that's going to be quick.
This one?
Yeah.
I'm down.
Ooh.
Wow.
Ooh, I think you might have got it.
Yeah, I see a flash of silver.
Okay, Roger. I'll hold here.
Sit down there.
Sitting down.
Yeah, this one. Yeah, I see it.
Yes. Well done. Okay, that works too.
Okay, see, that's where I want it.
There you go. You feel better now?
End of the,
The question is what to do now.
Yeah. Yeah, it's not in the bag till it's in the bag, but that was a good catch.
We're gonna put this in a bio box. Is that correct?
I think it's tricky because I don't think the whole net can fit in.
We modified it.
Oh, okay. I thought the net looked different. It's got a new trim.
It's a new digs. Okay. While you're there, I'm gonna pour it out.
Just go with the camera.
Roger, sorry.
Ah, it's in there.
Oh, you
Yeah,
folded
very
it
nice.
over.
Yeah.
Sneaky. That's awesome. Alright, portion out.
Alright.
Okay. Yeah. Alright, and I think it was B2, I think. Rocking in a bit.
Yes.
Yeah. Edge of the ring. So that's going to
BB2-B, or not 2B. If we're lucky.
Nice.
Box gonna close. Pressure.
Let's try it. I think it's, yeah. It's a little lipped open, but
It's nicely tangled up.
Good fishing job.
good fish and club.
Well done, all.
So what's this part though?
Well, it's the one I wasn't sure about, so we're going to find out now. I can get a good look at it.
Alright, I'll go chase it down to the south right now.
Yeah, I'm aware that they mentioned an iceberg and then we got distracted.
And the iceberg and
What happened with the iceberg?
Check in with Bridge, screaming. So the net's good, but it's a one-shot device.
There was a question in the chat about the red color of this poop. If we are correct in our assumption that this may be produced by the crab eater seal, then the red color will come from the krill that they are eating. Now the question is why the krill produces this red color. It may be that it is getting from the plankton, but the crustaceans, they can produce their own carotenoid pigments
Anyway,
as a protection against UV radiation. Also, the lipids of many crustaceans are red. So it may be a production from the krill itself, or it may be indeed accumulated from the plankton. In saline waters, there is some plankton that produces red pigments, and this is incorporated then into
So
consumers
like-
like flamingos or crab.
We had our fun.
yeah, they're fun and I get those.
Here we see a sea pen. Anemone.
Bipod fish.
Bipod fish, a big one. Oh, fall it down.
Yeah.
Come on. Yes.
I'm determined to get that name to stick. So, it wasn't a bipod fish, that's just what we've been calling them, but that's the ice fish, or sometimes called the crocodile ice fish. That's the cool ones that don't have any blood pigment and full of antifreeze and all sorts of interesting biochemical things going on with them.
So we're a group here, we went.
Crinoid.
Big crinoid, big sponge.
We've seen some bigger, older looking things coming in now.
Yeah, that's true. Yeah, there's a number of big sponges.
So we're coming up on waypoint 7, and we have another rock request at this waypoint. And since they're rather sparse on the ground, I'm going to allow it. We can start looking early.
Maybe we find the green sponge, rock, sorry.
I've been given a white whale. I've been given a quest.
ROV Greeks, continuing to waypoint now.
Roger.
So they wanted a pink rock for a while, and then we got a pink rock, or at least the team before our team got a pink rock. So now if we want to even the score, we apparently need a green rock. But I don't know how sincere that was. I don't know if there is a green rock.
I don't know.
Not really sure on the age of some of these glass sponges. They can certainly grow reasonably quickly in optimum conditions. They tend to grow quite quickly and then slow down once they reach a certain mature size, and then they start diverting all their energy into reproduction rather than growth. But these could be, in the hundreds of years and the decades. But we're not entirely sure. Part of the problem is that they feed on such old material. It's hard to sort of date them because they take on the characteristics of the old material that they're filtering and eating. It's quite an issue in the deep sea and in polar environments because these circulations can take so long, and these animals are feeding on sometimes very old material. It sort of contaminates the aging process. Might hear some noise. We're just swapping out the ROV pilots. Despite how sort of soothing and therapeutic this can be to watch, it requires like really high levels of focus and a constant awareness of where everything is in 3D space. The vessel, the vehicle itself, the manipulator arms, and the cable coming out the back. So it fatigues you really quickly. So a bit like air traffic control, we have very regular swap outs of the pilots, give them a chance to rest. Quest for a rock. So if you see a good sized rock.
Quest for a rock?
We're on a quest for a rock.
You got a color preference?
Well, green.
The green one would be good.
Green?
We head over to our expert going to waypoint 7 now. And you guys got a fish and biobox in a net though.
So we can open that.
Yeah, open that.
And these are the icebergs making us go south?
Yeah, we dodged it south and then now we're just
So now we're past it already.
going back to the thing. Okay, have a nice.
don't know if it's just by chance, but I've seen a few of the notothenoid fish now sitting on top of a sea cucumber.
Oh,
There was one back there that was sort of sitting under one. They might just treat them like a rock. They don't move very quickly, so it might just be a nice piece of cover.
yeah.
But what are the odds of sitting on top of one? Thank you. You're just reading my mind. I think I recognise it. Yeah, I know what that is. Yeah, it's the ones that like to donut up. Oh. See, there's sort of a gelatinous envelope around these fish.
Look at this minute sea cucumber.
Ah, that is tiny.
That's really, really tiny.
Mini pig. That fish is only about 10 centimeters, I think. A little longer, maybe. That's what makes these visual transects difficult. As soon as you slow down or zoom in, you see a million things. That's what layer do you want to go to?
So our shift is coming to an end. Thank you for watching and listening to us. Now Sophia will take over. And we will be back at 4:00 p.m.
And the octopus on the left. And the predatory ribbon worms. Maybe sneaking up on that sea pig. Or maybe that's the back.
Is it like more pink than usual?
Maybe.
No?
Maybe. We've seen some little piles of red pink.
Yeah.
The crab eaters, there's a bit of red.
Yeah.
But yeah, pretty consistent. We've been finding it hard to find the rocks.
Yeah, I suspected so much.
But we've gotten there.
But we put them there at locations where we can follow the terraces.
Yeah, but you can only be like you can be a meter over and not see it.
Exactly.
We got one Sakito, a couple of rocks. There is a fish in the net in rocks too. I'm thinking this is a leftover.
Yeah.
Just in this
no,
template.
This is, yeah.
No
This
worries.
is just when it drops out there. So
Okay. It is in a net. It is a feisty fish, but it's fairly well tangled. So
okay. Preferably not opening.
not leaving it open, but I think you're safe to put things in. I don't think it's
For
going to
now,
get
we
out.
can use this one.
Yeah, but no, it's well thought. Yeah,
Interesting
that's
fish,
good.
right?
It is because I'm not sure what it was.
Okay, there we go. It's easy.
I think I have a good challenge.
Thank you. Have a good rest. Good morning, guys. Plus 1.7. Okay. Okay.
Bridge, ROV Bismuth.
ROV Greek, sorry, say again.
Got a new waypoint for you.
New waypoint.
How are we looking on ice? Do you know, Zach?
Are we looking on ice?
Yeah. For the next waypoints.
Bridge, ROV.
Yeah, ROV Greek.
How are we looking for ice and stuff on our next waypoint, that waypoint 8 there?
Yeah, it's looking good.
Looking good?
Okay, thanks, sir. Looking good.
Thank you.
This is like a weird angle.
It is. We can turn a little bit. It's okay. We don't need to be exactly on the line of the waypoints. Oh, there's an octopus there. It's cute. Looks different from the others we've seen. Can we zoom in quickly? Oh, that's very cool. Thank you. Can we go slow here just to have a quick look? Okay. Oh, can we have a look here on this? Go a little bit around to the right side or to the left side of the rock as well. Thank you. You can continue. Oh, sorry. Can we just have a quick zoom on this one? Oh, that's nice. It's a snail with a buddy.
You want that?
I don't think so. Yesterday we did get one very similar to this.
Snail?
Yeah, so it's a gastropod. It just has a buddy on top.
Huh.
So you do see like
That looks weird, huh?
the feet area, here, and then the shell is here.
Okay.
It's just completely covered by the anemone. The anemone. The word that is very difficult to say. Yeah, we can move on.
Good morning, bom dia, bonjour. There's many languages online today. We are picking up from Pedro and Tom, the amazing Pedro and Tom. They're incredible at narrating. So it's Sophia and Andres here. We are still in the deeper, well, going slightly upslope now. This is part of the second dive, if I'm not mistaken. Yeah, second dive on the location outside the area we explored during the last week. The expedition, as part of the expedition Climate Connections at the IC interface, led by Dr. Patricia Skete and Sasha Montelli. This expedition is also under the flag of the Challenger 150 initiative, and we have been mostly spent most of our times in this area close to the Ronne Entrance as part of the expedition. I think this might be the last dive in the area, and then we're gonna explore a new station. This dive on an area exposed for longer term, at least for a longer time, outside ice, might have different productivity regimes, different circulation patterns. So that's why we were interested in coming to this location and double check what we do see on this area or not. We are going again from a deeper site, about 800, to a shallower location, much shallower than we were yesterday, about 200. Again, through different topographic features to see if we can grasp of sedimented areas and rocky outcrops, different orientations to the currents, to have as much of an overview of what it is living in these locations. I'm going to switch to my language and hope there might be some Portuguese over there as well hearing. Bom dia. Bem-vindos ao segundo mergulho nesta nova zona. Esta zona tem sido, já agora comparte a expedição Climate Connections at the Sea Ice Interface Cruise. Esta expedição tem estado maioritariamente focada na zona da Ronne Entrance, no Bellinghausen Sea, e estamos agora no o que nós pensamos o último mergulho nesta estação, e o segundo numa zona onde tem estado mais exposta, pelo menos já durante mais tempo, numa área onde não tem tido cobertura de gelo permanente, o que nós chamamos de ice shelf. Temos novamente uma zona, começamos numa zona mais profunda, cerca de 800 metros, fomos até a cerca dos 200. Não tentamos cobrir o máximo de estruturas geológicas topográficas possíveis, quando tentamos também orientar os transects numa tentativa de observar diferentes áreas expostas a diferentes correntes, orientações de correntes, para tentarmos ter uma melhor observação, digamos assim, dos animais que vivem nesta zona. De seguida a esta área iremos em princípio para uma nova área, mais fora, portanto completamente exposta a condições sem gelo, digamos, permanente, e então aí esperamos ter condições diferentes para podermos comparar também com esta área, e claro, os colegas também podem ter, inclusive terem fauna diferente para poderem descrever novas espécies ou confirmar a presença de espécies que existem. Vamos continuar para já com observações gerais, apanhando animais que pareçam ser novos para as nossas observações. Can we stop here so we can have a close look? Can you have a look again on this kind of yellow patch? I think we haven't seen much of that. This seems some kind of encrusting sponge. Or bryozoa, I'm not sure. Thank you. Andres, if you see your rock, this is the location for a rock. Okay. But that one in the middle I think might be too big. Yeah, they're in the corner, yes. This one.
I think the one next to it might be good.
This one, Andres? Just below? It's not too big, this one?
No try.
I can't see the laser. Ah, yeah, it's too big.
It's slightly bigger than the laser. The other one.
On top?
Yeah, the one on top. Is it stuck?
Yeah, but I think I knocked it loose. Yeah.
It's the challenge of the morning.
Yeah, I think they're pretty good. Clear.
Let me see a video of the wheel from yesterday.
Ah, yeah. Well, Tony actually took a better video after, so I'm going to show you Tony's video. This one. That was very cool. It was just.
Yeah. So what type of wheel do we think that is?
I don't know. My fun was a minky, but it's quite small. I don't know. I'm no expert on the wheels. Let me see. I also had my own at some point. I took way too many. Here. It's here, is it? It's fun.
Yeah, I was up on the outside lounge and I heard it and I saw it, but by the
you
time I
saw
got my phone out, it was gone.
Yeah, I did saw it more times than I could film it, but I think it's more worth to have a look than rather film it.
Yeah.
They have been around, and I can see why. The whole seafloor is pinkish, so it's leftover krill. So I'm sure the surface is full of food for them. They have been enjoying. Yeah, we're very lucky.
Bridge RV, you could come to no stop please.
Full stop.
Thank you.
Yeah.
Good morning.
It's good morning. Can you say it back? Hey, you have to say it, not me. Is stuck or?
No, it's there. You can see the macro.
Ah, okay. have just a quick zoom before we put it on the box? Yeah.
Mirror, okay?
Okay, that's great. Thank you.
Thank you. We can put it on the outside rock basket.
Okay.
Just basket one.
Are we doing anything else here, or are we done here?
We're done here.
In that case, let's take off, and that way we can
Okay.
See better.
the basket. Like a day.
Mhm.
Okay. So,
Yep, thank you. I'd like to have a quick look at this rock here in this area. Look at those beautiful rocks on this. Down there. Thank you. I think it's good. What about vertical?
Turkey. Turkey.
Oh, can we have a quick some more a zoom in this? Some kind of priozoan maybe, but I wanted to double check. Thank you. There are again the little sea pigs.
What's it? Yellow.
Thought it was a priozoan. Maybe. Thank you.
What's that? AM on the winch.
Can we have a quick look at the sponge? Yeah, that's beautiful. We have seen them, but sometimes they have also interesting associates. Yeah, not the amphipods I was looking for. Thank you. Oh, and we are back to flat. Damn it. similar to the location we were just yesterday. The sediment areas are full of sea pigs from the family Alpididae, and some very funny sea urchins covering themselves by hats of leftovers scallops. We do see a lot of marks on the seafloor as well from their activity. Some tubes of polychaetes once in a while, like here. And the interesting thing is that they seem to be the ones mostly profiting from whatever these pink patches we see that fall from the surface that are basically krill leftovers from the feeding activity, or that krill, of course, that just sinks down. There's, of course, also many smaller organisms that we are not able. We can do that. We don't need to be exactly on that waypoint. There's, of course, many smaller organisms that we do not see. Some are almost visible when we zoom in, like the mini sea pigs. But there's, of course, the tiny organisms that are living in the sediment, that they do have their own activity and they do have their own job. For example, the macro and the meiofauna, so organisms like the polychaetes and the little crustaceans that live in the sediments, the clams, the little clams, bivalves, and many others. The meiofauna, where nematodes and copepods are usually the most common species, or the common groups, not species, but they have their own role, and they contribute, for example, to recycle this organic matter that's on the surface, bring more oxygen down on deeper layers on the sediment. So together with the sea pigs that you see here, the little brittle stars, the sea urchins, they all kind of work together into making sure that this organic matter is processed and available for different trophic levels. The sponges here around as well, they're great filter feeders. There's some incredible, yesterday I was showing one of the geologists that was asking how they feed, and sponges are incredible filter feeders. They and powerful, actually. So I was just showing a video on the internet where they included put some ink at the base of the sponge, and very quickly you see the whole ink going out from the main opening on top of the sponge, and that's how powerful they were. So it was just a couple of seconds. So of course, then when we go to these rocky areas, then you have some other kinds of animals. Oh, and of course you have these nemerteans, which are predators. The anemones can be both, depending on, they can actually fluctuate between depending on their needs and the species, of course. And on the rocky outcrops, you do have different communities as well. The fact that these are mostly mobile species allows them to go and search for the food that they need. Even the crinoid we see right now on top of, I think, maybe either a sponge, a dead sponge, or an old berry covered rock. I think it's a dead sponge. Yeah, it's full of brittle stars as well. They can also swim from one location to the other, so they can filter the particles in the direction that it's best for them. And yeah, you have the whole kind of different strategies that allows these communities to thrive and be here for a really long time, for sure. As we see, these sponges are huge, for example. This indicates that at least they're here for a long time. We don't know how much. We were discussing a bit that yesterday, and we're going to ask a couple of experts if they can help us with that. But yeah, we think this is quite a happy community here. because yesterday, for example, we did saw the sea pigs we collected had eggplants. I'm gonna double check. We'll say yes.
So how do you tell the difference between the males and females if they do?
I would say internal organs. Yeah. Let me double check that. I'm not a specialist on sea pigs. There's also a lot of species that are able to switch depending on the developing phases and also the environmental conditions. And they have kind of big cycles. Oh, there's the bipod fish. It's actually from the Antarctica. And apparently they do synchronize their reproduction cycles to the productivity peaks. So when you have more food sources, like a spring bloom, for example, which here is from December to January, they tend to, that's when they have their spawning events, or after that, basically. So after that productivity time. That's very interesting. Always learning. It's a sea pen. Can we have a quick zoom? They do have those amphipods that are attached together. Damn it. So these sea pens, they have some amphipods that are always attached to them, but they do not go away. We cannot pick them with a suction sample at all. So you have to pick up the whole.
Yeah, so they
yeah, and even in the lab, it's tricky to pick them.
Yeah.
Patricia was kind of putting another obstacle so they can move themselves to the forceps, and only then she could separate them from this little software.
We got two the other day and we put him in upside down. Is that the ones you're talking about?
Yeah, yeah.
What holds on? It's got like a tentacle or something?
Yeah, they have kind of some paws that they can attach very well. You see them? But they were super well attached. They really didn't want to go away. Even here in the lab, so they were still quite strong. Yeah, thank you. Oh, and there's a dead one on the seafloor. Oh.
You want it?
No, not now. I think they're okay. For now, we're good.
Right.
Thank you. But they're super cool. Well, they deserve to stay if they're hanging that strong. that one is by itself. Xenophore. Cum jelly. Doesn't look very happy.
ROV Bridge, you happy with the ship continuing to the waypoint?
Yeah, we're gonna turn.
Yeah.
Yeah, we're turning now.
Ah, okay, it's back to activity. There you go.
Focus there for me, sir.
Oh, that's beautiful.
ROV Bridge.
Go for ROV.
Yeah, you happy with the ship continuing to the waypoint as we are at the moment?
Yeah, we're turning right now.
Copy that.
Thank you.
Right.
Go southeast, right?
Yep. I'm gonna turn that in now.
Another one. Okay, oh, nice one. Oh, can we have a quick zoom on the Antomasis, the mushroom coral? Thank you. Thank you. That's great. It's a couple of them. It's interesting. Yeah, we can move on. Oh, it's a nice cup coral again. Nice. Oh, that's beautiful. Can we stop here?
Yeah.
I think we might have a quick look at that coral, and if it's what I think, we might collect it. So, oh, look at the sea lily going away. We're bothering them.
Oh.
Yeah.
It's gone.
Very fast.
Nice. It's a squid day. Remember? So Tom just sent us a super nice video from the light lures with a squid kind of being annoyed at it. Oh, that's nice. Yeah, we definitely would like to have a good zoom on these corals.
Yeah.
Yeah, that's what I'm thinking.
Looks weird.
It looks no, yeah. It does look like an old yellow colony full of zoanthids, but
Bridge, ROV.
ROV bridge.
You can come to a controlled stop around there.
Sorry, you're very quiet on your last say again.
You can come to a controlled stop there.
Okay, come to a stop.
Is there like a couple of meters on the number of that? 10 meters.
10? 85.
Yeah. Okay,
Yeah.
it's all yours.
so I'd like to have a good zoom here on both this one, what is below and the top part. So some good shots to have a good look. Let's just wait a bit so we can focus. I like to focus first on this one.
Focus now on this.
Thank you. And then the one in the back. Thank you.
Which one do you want?
And there's one below that I think is more whitish. There, just there. Yeah. I'd like to have a look as well. This one here. Thank you. Okay, so we would collect both a little piece of this one
Mm-hmm.
and a piece of the other part. And I think,
Both of the yellows.
yeah, a little piece of this one here, and then
Big one?
a piece of this one.
Where's
Yeah.
this going to go in a quiver?
On a coral quiver. Well, two coral quivers separate.
Let me come over with my walk around. Okay.
There's nothing on any other one.
No.
There's only one. No, there's nothing on the coral quivers at the moment, so it's good. It's free.
Which one you want for the big one or the small?
We can go first. Yeah, it doesn't matter.
I'll go for the big one first.
Okay. So a piece about the laser size.
10 centimeter.
Yeah.
Please.
More or less. It's like not very specific. It's actually supposed to work okay.
What about like that?
Okay, yeah, sure.
You gotta get the entire thing.
That much.
Okay, that much. I do think these are entities, but I want to confirm.
Yeah, you got it.
Okay,
And you're
thank
gonna
you.
get
Some
squid ink.
squid slime.
That guy's not happy with us.
No. Well, he's right.
It's in the app.
Can I have
bring
just
it up into the
a...
darkness on the glamour shot?
Yeah, exactly. Like you say, bring it to the darkness. It's like very dramatic, guys. That's great, thank you. Thank you.
Okay. Glamour shot complete.
Thank you, Zach. Yep, that's good.
Oh, it broke off.
Uh-oh.
Good job.
Did it broke off completely?
Yeah. No.
Not the other one. Yeah, okay, good. No, just confirming. If it did broke up, we'll put the whole thing there.
Yeah, bring it up to the deepest darkness.
That's right, Mom. Photoshop.
Higher. There we go. Okay.
wanted without the floats. I didn't stay awake for that. I have to say.
What is 5 or so?
I was doing dive planning, and then I went to bed. Oh, can we have a look at the sea star there? A person has to sleep eventually, you know. Eventually. Oh, nice. Sun sea star? Yes, I don't know.
So is that different than a sea star and a starfish?
Starfish, sea star.
It's the
I don't
same?
know. It's the same, I think. It's a language thing, isn't it?
There's a famous paper by Christensen et al. I think 1956
Mm-hmm.
that specifies why this is not a fish, so it's a sea star.
It's a sea star. Okay. There you go.
There you go.
Yeah, we can continue. It was just for a quick sun. It's the sun sea star in this case.
There are no more starfishes.
We did had a cruise where we were logging different people. It was a German vessel, and of course there was different nationalities, and overnight you have people logging, etc. And then there were a lot of now I have starfish in my head, but sea stars on the seafloor, and we had this immense discussion on what it was. And at some point people would just call it in different languages, so there was no confusion. Thank you. And I think we can go to waypoint 9.
Yeah.
It's just having a quick look at the map, but after 9-10 we go kind of upslope, quite steep. So we should...
Probably started asking by then.
You know, I want to let you catch up a little first.
Should be alright. No.
You got a
Okay.
hard
Yeah.
sonar turret coming up on your left a little bit.
Yeah.
Probably just a big rock.
Mm-hmm.
What those remind me of? You know, the movie Alien.
Really?
The eggs.
Thing jumps out.
Yeah.
Oh, really, guys?
Yeah, it looks just like that. You know,
Okay.
James Cameron stole a lot of
Deep sea stuff.
Yeah, deep sea stuff. And put it
It's
in
true.
movies.
Like from Nemo.
Yeah.
Yeah, that's true.
It looked,
Nemo's inspired the alien, didn't
I didn't know that.
it? Because they hollow out the salps.
Yeah.
They live in the middle of the salps.
Okay.
he used that little, what was it? The crane wing? The flying things. He used
Okay.
that in Avatar.
Ah, true.
Yeah,
That's true.
The little kind of white thing that was flying around the
Yeah.
trees. Sure, you
Ah.
took inspiration from that.
Now I'm thinking, Zach, you're right. The the amphipod for the alien. Yeah, yeah, the amphipod.
Do you know there's actually a movie about killer tardigrades?
What? No way.
I don't recommend it. It's pretty terrible.
I bet.
I feel like a really bad movie.
I do like really bad movies, especially on a Sunday afternoon.
Like Harbinger Down.
I think Sundays afternoon are made for bad movies. It's like lazy moment.
What happens is the Russians sent tardigrades to space.
And they become huge, I imagine.
Well, something. Yeah. Evil, let's say.
Aw, but tardigrades are so cute. Oh, this is a sponge ground. Nice.
Maybe I do a still just that one.
Sorry? Yesterday we also had this massive black darker sponges
Mm-hmm.
like this, but quite
Bridge,
tall.
ROV, got a new waypoint for you?
I think those were good.
I don't think so.
Updated waypoint.
It was just a coloration difference. I'm gonna send them to a colleague. Yeah. I'm gonna send them to a colleague. Maybe she can help us with identification. Oh, there's the snail again with the anemone. Can we pick it up? This one?
You want it?
Yeah. Just to double check if it's, because here we cannot see if it's the same snail or not. We only see the covered anemone. I know. Does it have a snail?
No.
Nah, this one is just an isolated anemone. Okay, good.
So you don't want it?
No.
It gets to live another day.
Just a spongy anemone? Anemone? In Portuguese, anemona. Simpler.
Rocks coming up.
Yeah, we can have a good look at them. Okay, check here. Saw something pickish. Yeah, can I have? I think we're gonna collect this.
Okay.
I'm not sure if we've seen this one or if we collected this one, but it's
Suction.
yes, with such a sample here.
Which jar?
We can put it on 4 or 5. Either is fine.
I'm too far away. I mean, lo mismo de batir la probably.
Can we just have a good zoom as well?
Yeah.
Yeah.
What's that?
We'll have to wait for a little Liz to come back.
But Zach is ready, clearly.
What jar did you say you wanted?
4 or 5? Well, it can be 4. It's there already. Yeah.
Stop them moving or leave them moving?
I can stop that. I don't know.
Bridge, ROV.
ROV bridge.
Hey, can we pause that move? We're gonna wait here and take a sample here.
Yeah, no problem.
Thank you.
So Andres found this.
Top up.
That's a real one y'all saw? That looks like Photoshop.
It's a 3D printed hat. You can see the lines of the 3D printer.
Oh, they made that. That's
Just-
on this boat?
No, that's a coastal sea urchin, so it's definitely not on this boat. But it's an idea for you.
Yeah, it'd be funny. Except I think somebody will probably complain about putting plastic on the water.
Yeah, I know.
The sea urchins wear hats anyway.
They're hiding from predators.
Yeah, it's fashion.
It's just a fashion, deep sea fashion then. Okay. Why not? can see it.
Okay.
I think so.
We're on the right jar for?
Yeah. But I'd like to have just a quick zoom so we can see it.
Sure.
How does it look in its natural habitat just before we collect it? Sometimes the coloration is a bit different. They do arrive pretty good on board, but still.
Take a look at the default number.
Mhm. If you're wondering why we are stopped at the moment, basically we found one of the target groups, the Peracarids, an isopod that we are aiming to sample. When these locations are verifying sediments, so that means every time the ROV stops on the ground, we might have a lot of particles suspended, and then we have to wait a little bit. But it's totally worth it because this is one of the main targets of this expedition. We have experts on board that are taxonomists that describe the species. Actually, the head scientist of this expedition, Bedesishkit, is an expert on these groups, and she already found some interesting findings on some of the stations. So every time we do collect this, we do have to collect the whole animal because they're quite small. But we have been finding already some interesting. There's a potential interesting species that may or may not be new. That's have to be confirmed in the lab later. And then, of course, more have to be studied and to verify both with morphological identifications and molecular tools. And once that is done, then we can at least understand if the species distribution range are expanded. For example, if we have species that are going way further south, for example, than initially predicted, or we just have we have new species, which is pretty amazing in these locations since they are quite isolated from due to the water masses around Antarctica. So it depends a bit on the group we may find. But yeah. Oh, there's another squid. That's cool. You guys may not see it on the main camera, but we do have them squids.
Are you happy with the vision?
I'd like to clear up just a little bit more, yeah.
You want me to? I'll do that.
I'd just like to see it.
That's a read all of it. It's clear now.
Alright, it's clearing now, yeah.
Squid.
Still there.
The same guy's been with us. He's like really mad.
It doesn't like us, huh? No, I think we can collect it. I think it's gonna be quite messy anyway.
Okay.
Yeah.
Suction is on.
There it goes. In the end, though,
shoulders up a little bit.
Come on, little dude. There he goes. Thank you.
I'll just keep hold of this in case this is something else.
Mhm.
Close lock.
Okay. Thank you so much. Okay.
Yeah, we're looking at the pilot.
There's a squid again. Doesn't like us. I'm gonna say it's a she. What is the name they're calling the squids on board? It's Jorge. Okay, but for this purpose, we're gonna be Jorgina. Georgina.
Right?
Georgina.
Is this good?
Yeah, it's a
Why?
squid. I don't know. That's what. Who was it? Who called it Georgia? You did? And what's the female version of that? Georgina.
Francisca.
Francisca. Oh my god. So that was Francisca then.
But the search is Margarita.
The sea urchin is Margarita. Okay. Sorry, can we have a quick zoom here? I think they're polytubes, but just want to confirm.
Bridge RV, resume your move.
We can have a look at these ones or there. It's fine.
Copy that. Continue your move.
Careful, something. No polytubes. Yeah. We seen them yesterday. I think they collected. Okay, thank you. We thought at some point it could be forearms, but it's strange. There's a nice octopus ahead.
Octo.
Yeah. Octavia. Oh, that's beautiful. Look at the colors. Oh, there's two. Oh, that's nice. It's Dumbo. There's Dumbo and another one.
Dumbo's to the left.
Yeah, it's the purple one. Nice. Thank you. Yeah, it's too sedimented. No worries.
It's just look bad.
That's what it is. But that was very cool. Is it possible to orientate a bit so we are more in line of with the slope?
Yeah.
It's okay if we go a little bit off the path itself, but so we can have a better orientation to the slope. Yeah, it's fine that we do not go to the waypoint 9 exactly. Yeah, it has amphipods again. Nice.
You gonna cut the corner here at 9 and 10 maybe?
Yeah, we could do that exactly. That's okay.
Maybe I should move it to the
Yeah.
south.
Yeah, that's good. I think we're in a good
Ready,
area.
ROV, up to your waypoint.
Take the waypoint, thanks, sir.
I think you're on a better orientation this way. Otherwise, we would be quite on the side. That's for Tom. Yeah, we can move. Yeah, otherwise. Can we go slow here so we can have a good look at the areas? I'd like to do a good zoom in on these first sponges there. Oh, wait, we want that before the sponges. So I would like to stop here. Have a good zoom on this one. Yeah. Wait. Yes. We can put it 6 or 8, any of them.
6 or 8?
Maybe 6, for example. hunting for a polychaete, yes.
It's moving, but there shouldn't matter. It's just gonna go across, so it shouldn't.
Mm-hmm. So we go towards more waypoint 11, I guess, in this case.
Good eggs?
Good eggs.
What kind of eggs?
Just
Crumbles?
sunny side up ones.
Ah, okay.
And hard boiled, of course.
There you go.
Coconut chocolate.
Oh.
With coconuts?
Coconut, like butterscotchy.
Oh.
It was quite good.
Yeah, no, I don't like coconut.
What?
I know, it's weird.
It's possible?
It's possible. I'm also the worst Portuguese in the sense that I don't like olives and I don't like tomato.
You don't like olive?
No, I do love olive oil. But yeah.
You like cod? Or you
I
like
love cod.
Okay.
No.
That would be very odd.
One of my best friends does not like cod. So that is odd. Yeah. So cod is the Portuguese main food. That and pastéis de nata. You like it? How not to, huh? And they have the perfect size. Perfect size in the coffee.
Do you call pastel de nata or pastel de Belém?
No, pastel de nata. Pastel de Belém is from the precise place. It's from Belém in Lisbon. Otherwise, it's pastéis de nata. Because it's from that bakery, very old bakery. Do you know how it looks? No idea? Oh, I'm gonna show
But
you.
I will not.
No. It does look like this.
What is it?
It's kind of a little pastry cake, something like this size.
Sweet or savory?
Yeah, sweet. So it's massa folhada. Oh, in English.
It's like,
Massa folhada? I don't know.
what do you call it?
Like the croissant
Like
paste.
croissant,
flaky.
Flaky dough. And then inside is like eggy kind of filling, egg and sweet.
Amazing.
The fat part of the milk, which is the best part.
Yeah. People call it the little fun. We hate that, but it does seem like that. It has the perfect size. And very often they sell it in the coffee places, like a coffee and that together as a pack. Yeah.
Holy water.
We park up in Seafloor. Grass on Sharpello, polychaete is it?
Mm-hmm. Yep.
Yeah, right in front.
Should be just there.
So we'll keep it straight.
On that. So you have a good breakfast. Good morning. I hope it didn't run away.
I'm sure it's busy.
At this time, no. At this time, it's never busy. That's why I love it. It's way more relaxing.
Ship's currently moving, but it's. So the ship's currently moving. You can pause it. It's just gonna go here.
So basically, instead of going directly to
Alrighty,
a
Rich.
person,
Go ahead.
Yeah, we can't continue moving in this direction. We've got enough
continue in this direction until you're ready.
Okay, if you want, we can put an all stop here, if that is okay for the ship.
Yeah, okay, we'll come to an all stop and yeah, let us know when you're ready.
Okay, great, thanks so much.
And then we don't need to go to waypoint 10, we can go directly to waypoint 11, if that's possible.
Okay, yeah, it'll go directly astern, so it'll probably be, might be a little bit to the north.
Yeah, as we go. It doesn't need to be specific, but again, exploration mode, you know? It's like, we try. But it's what is possible. Oh, it's snowing. Nice. Clearing up, damn it. They usually do not help. They make it worse. But it's a worth waiting, let's say. So. Let's go with us.
We are on an incline. How are we?
A little bit, yes. So there's just a couple of rocks just in front of us, like a meter away. But the polychaete should be right there.
Pulling. Seems to be pulling due to some sunlight. It's not, I don't have much trust, but it seems to be.
Mm-hmm.
Yep, it's not clearing now.
Yeah,
No,
it's
not at
clear
all.
enough.
Just above the back.
I'm not sure. Do you want
Can
to wait a little more longer? Or?
Can we try pick up the suction sampler, go a bit up, and see if we see it as we move?
Do you want to pick up the suction sample?
So we can grab the suction sampler in the hand.
So it's
Sitting
in
on
the
the-
hand right now.
Ah, it's in your hand. Ah,
Yeah.
okay, yeah, perfect. I was just thinking going slightly up.
It's starting to actually settle a little bit.
Oh yeah,
See what I
maybe. Yeah, it's true. As we change plans, of course. It was quite a big polykit, so if it didn't run away, it should be easy to spot. Again? There has been squids hitting us for a while now. Carlos, is it possible to have access to that video feed? Do you know?
This one? The
Yeah.
average one? Yeah.
Yeah? Because Tom was very interested in that. Don't see it yet.
Was it in the center or on?
It was in the center, yeah, but it's a mobile animal, so it can kind of go away. So wondering if it's not a rock. I think there might be something here. I think that's it, yeah. Yeah, that's it, yeah. Mm-hmm. I think that's a polykit. Because there's a lot in here, but here, you know, I can just here. Yeah, that's a polykit.
Oh.
You see it? So we can go for it. I don't think we're gonna have a very clear image anyway. And we did see it as we were going up, so it should be fine.
You want?
Oh, in front of there.
Where is it? Here. Yeah, it's here. It's not the clearest possible, but it's here. Don't know if that helps. Yeah.
Are we lined up on the right track?
Right.
Yeah.
6.
We're good? Oh, it's good. Yeah. So let's see if it's in. Bit messy. Yeah, it's there.
Anything else? Or else it's,
Yeah, now it's inside, yeah. No, just that. Thank you. Just before we move, can we try to see if there's clearer part here on the rock that we can zoom in? I don't think it's going to be very clear, but just to have a view without moving too much sediment again.
Really?
So there's a rock just in front of us up here. Section sample 6.
6 or 4?
6. No worries.
Not the point.
The 4 was the isotope. The politics is on 6. No worries. We can edit that afterwards. It's okay. It's too messy. It's okay, we can move on. No worries. It's easy to change the numbers. Towards waypoint 11.
Bridge ROV?
Waypoint 11. You could proceed in that direction.
ROV bridge.
Hey guys, okay, you can go ahead and proceed with your stern movement to avoid the ice. And we'll just follow.
Copy that.
If we still see somewhere closer to 10, something interesting on the sonar, we can still move slightly. Some rocky outcrops or something like that. Like the waypoints are not fixed. Any coordinate? No worries.
You know, I don't go back to the names of the crops.
No worries, I'll make some notes. There's some tunicates, sea pens, oh, there's a lot of stuff. It's nice. Sorry, can we stop here and have a good look at this stocked? It's fine, it might be too much sediment again. I'd like to have a look at this. It's a tunicate, but it does seem to have some zoanthids with it. Yeah, thank you. It was just to have a quick look. It was just to have a quick look. We can continue. to stop here and take a saquito, a little bag.
Obviously, we're just able to do that since we're passing the basement.
Oh yeah, true.
Bridge ROV.
Go ahead, RV.
Hey, they're looking for a sample, but are you guys able to stop right now since we're woody ice?
Yeah, no, that's fine. We can stop.
Roger. Thank you.
No particular, just in this area, so it's fine. It becomes coarser, so I think it's interesting.
I'll take the second out.
We can use number 2, so on the outside basket. This says at the end of the list, you put other animals. Yes. That's it then.
But that's a sacrifice with sediment.
Well, it's animal purposes. It's okay.
Usually sediment with animals or animals with sediment?
That's the question. It's like, what comes first, the egg or the chicken? It's very tricky. Who knows?
Number 2, right?
Number 2, yes. The other one was taken already. If we can move just slightly back, there was more green. See more green, more slightly small rocks on the seafloor. Or to the left, I don't know if it helps with the green.
That kind of big rock, maybe go behind you.
Yeah, we can go behind. I was just looking for that mix of soft sediment and a few rocks, small rocks in there that we just saw. Yeah, here is good. Anywhere here is fine.
Yeah, it's good.
Yeah. Yeah, it's perfect, yeah. So ideally mostly surface parts, not so deep. So it's like fine.
Something like this, a bit more shallow.
No, like that is fine. Yeah, like that is okay. Just the first like 4 or 5 centimeters, as we say. Usually it's where the animals are, so. Yeah, and then we do a couple of more passes to try to feel a bit more.
Where is it? That's the.
It's possible a bit more.
Yeah. I don't mind just trying
Just to leave Americo very happy with a lot of mud to save afterwards. Just a little bit more, yes, please. I think we're okay. Yeah.
Good.
Yeah, good. We can put it on the outside rock basket. Thank you. And we can continue.
Go ahead.
Hey Jason, you can go ahead and resume your
I can actually probably head towards that waypoint that you put in, so I'll give that a go.
Okay, that'd be great, thank you.
Can we stop here and have a zoom here? Yeah. Yeah, I want to sample this one. Oh, this one.
Bridge ROV.
But first I'd like to have a good zoom.
ROV bridge.
Jason, would you go ahead and pause and
Copy that.
If it's okay. An Afipod, I think. Afipod.
Perception charge ROV?
5. Oh.
Well,
Is that? Oh. Well, okay. No picking him. No picking him. There was an Afipod that just flew away. That's okay. You already have one isopod in the morning. No, it was an Afipod. This was. But we have one isopod for you already.
don't worry, you can sample a rock.
You can. Oh yeah, it's actually a location for a rock. It is. Sorry. Yes. I'm gonna have a quick switch. Já estamos bastante avançados, já estamos. Não fomos exatamente ao waypoint 10 por causa da orientação. Estamos mais assim a meio.
Porque por causa da orientação. Não estava
Sim,
nada
a
bom.
câmera estava muito lado, estávamos completamente lados, não conseguíamos. Mas tudo bem, temos visto o rock outcrops e o waypoint 11 também é aqui no sul, estás a ver?
Muy bien.
O Tom apanhou um peixe esta noite, está aqui corrido. Pode-se abrir, mas preferencialmente usando as outras boias.
How's that? Just the angle was bad.
I just figured out and click back. Pretty what I was doing.
No worries.
Or just like a...
You know what? You know, I discovered sometimes they need to be stuck.
Okay.
Yeah, that one's fine. Thanks. Sorry about that.
Maybe you can try any of these rocks.
Where do you want this stash by?
It's like you went around.
There's one in the
this one. Let's do the one, like top one, this one.
Perhaps that one. That one looks doable. In the rock basket. Any of those?
Roger.
That's it?
Yeah.
Patricia, do you guys looking to sample more around here? Gonna sample.
What do you mean?
Are there any other samples that you want in here?
No, we can continue.
Bridge ROV.
Go ahead.
Please proceed to waypoint.
Understood, get ahead of it.
Thank you.
You can zoom into that.
Huh? Wanna stop?
Just zoom in. Oh no, it's gone. By the time we zoom in, it will be too dusty. So let's just continue.
Muy buenos días. Nos encontramos aquí en una nueva inmersión del equipo del ROV SuBastian en el mar de Bellingshausen, en la esquina inferior derecha del mar de Bellingshausen, bajo la isla Alexander, en lo que es la entrada de la plataforma George Sector, ya que es una de las plataformas de hielo más importantes de la Antártica, concretamente la 8ª plataforma de hielo de la Antártica, y estamos en un área que hasta hace poco tiempo era cubierta por hielo. Estamos cotejando las áreas que fueron cubiertas versus áreas que han estado más tiempo descubiertas del hielo. Esto debido a una reciente fractura de la parte sur de la plataforma de hielo, ¿ya? Entonces estamos en esta última inmersión ya en este sector, una de las últimas, lo cual nos permite comparar ya sobre la variación entre la fauna que ha estado permanentemente por miles de años cubierta de hielo, no directamente, pero en su superficie, vale decir que están en los fondos marinos que están cubiertos de hielo y que no reciben aporte de la fotosíntesis de la superficie, porque siempre están bajo esa capa de hielo que puede tener hasta unos 200 metros de espesor en la superficie, muy cerca del fondo en áreas más someras y más lejos del fondo en áreas más profundas, ¿ya? Pero que igual no impide que en verano la luz del sol penetre y que las microalgas pueden hacer la fotosíntesis y a partir de eso se desarrolle toda la trama trófica. Ya han sido días muy que evidencian mucho estas comunidades, su riqueza, su diversidad de especies, ¿ya? Por tanto, para nosotros es un hallazgo y es una oportunidad poder contar con esta información y más encima poder transmitirla en vivo. En nuestro turno de watching está liderado por Patricia Schete de Universidad de Aveiro, Portugal. Quien les habla es Cristian Aldea de la Universidad de Magallanes, Punta Arenas, Chile. Está Sacha Montelli del University College London y Leslie Novoa de la Universidad de Magallanes, Punta Arenas, Chile también. Ya. En este momento nos estamos acercando a hacer zoom a primero que todo sale un decápodo ahí, un crustáceo, que son de las pocas especies de decápodos que viven en Antártica, que se lo ha llevado la nube de sedimento. Y también estamos haciendo zoom en otros animales como esta anémona y una esponja que se veía. También más abajo se observan tubos de poliqueto. Son grupos en general muy frecuentes en esta zona que está cubierta de este tipo de animales, muchos animales que nosotros conocemos como animales sésiles, que quiere decir que no se desplazan en estado adulto, ya se anclan al sustrato y habitan toda su vida en ese lugar. Lo que estamos haciendo hoy es hoy, así como los días anteriores, lo que hacemos son un buceo largo de navegación, de transectos de navegación desde zonas más profundas hacia zonas más someras. Que destacar también que este crucero, esta campaña, tiene por objetivo realizar una cartografía detallada, un mapeo que se conoce de los fondos marinos de las zonas a estudiar, y aparte de eso estudiar muchísimos parámetros biológicos, oceanográficos, ambientales de esta zona, de los fondos marinos más concretamente, ¿ya? Y desde el punto de vista biológico, que es un eje de la investigación, se están estudiando la diversidad de las comunidades, vale decir la composición de la especie en todo su alcance, parte de la ecología trófica, DNA ambiental, etcétera. Varios parámetros que con el pasar del tiempo vamos a ir dando a conocer. Ya han sido días muy intensos de mucho buceo y de muchas actividades. Algo frecuente que suele ocurrir y que nos ha pasado en estos numerosos buceos es que el sedimento, al ser tan fino, cuando el vehículo toca la superficie del fondo marino, se observa inmediatamente una resuspensión del sedimento, lo cual hace más complejo también el muestreo en ese punto específico.
There will be this one, but I want to see what is clinging on this one. It's gonna be very complicated, just keep going. There you go. I want to zoom into this guy, exactly.
Lo que se está tratando de hacer ahora es hacerle un close-up, un acercamiento a este anfípodo que se especializa en vivir anclado a estructuras como la de esta pluma de mar que se observa ahí.
No, it's okay. I will be. Just wanted to check out the genus. We are not gonna be able to make it to species, and I don't want to go late. Let's continue. Motivo por el que queríamos hacer el acercamiento a este anfípodo, a este animal en concreto. Es para más o menos estimar qué género puede ser. Muy difícil llegar a especie sin
tener la muestra.
Sin tener la muestra, efectivamente. Ahí tenemos un calamar que hace un aterrizaje en el fondo. Sí, me interesan esos animales que viven ahí. En este momento estoy estudiando una especie del mismo género en el Ártico y hemos observado durante esta campaña diversos ejemplares siempre anclados y siempre bien agarrados a plumas de mar. Diferentes especies, pero siempre a plumas de mar. Hay dos especies de ese género citadas en la Antártica, lo que se conocen de la Antártica, ambas en la península antártica. Entonces sí. Y también hay diversas especies de plumas de mar. Entonces también me interesa saber si estos anfípodos tienden a anclarse siempre a la misma especie de pluma de mar o plumas diferentes. O realmente les da igual, solo quieren ese tallo idóneo. De momento me parece que efectivamente son capaces de vivir felizmente y no tienen preferencia por una especie en concreto.
Aquí lo que se observa es un fondo sedimentario muy rico en conchillas de diversas especies o testas de erizo que se observan. Debe ser muy, muy rico en nutrientes. Eso también lo evidencia la presencia de varias numerosas holoturies del grupo de las periagoni o lermogoni, ya que están ahí alimentándose, que son holoturies también que tienen la capacidad de nadar.
Sí, hay bastante. Y aquí es curioso que vemos como una especie de prax
Sí.
bajando por la colina. No sé si será un animal o será, incluso puede ser una piedra.
Una piedra o dos piedras que se han
Dos piedras en este caso, que se hayan desprendido.
Sí.
Pero realmente la densidad de holoturia es bastante llamativa.
Sí.
Es lo que venimos viendo en esta zona. Nos contaban nuestras compañeras que trabajan en oceanografía física y oceanografía biológica, que estamos justo pasado un par de meses del bloom de nutrientes de primavera. Esto quiere decir que hace unos meses, superficie cerca de aquí, cerca de esta zona o por esta zona, ha habido un aporte de nutrientes grande, una reproducción y una producción primaria muy grande por parte de microalgas, de un crecimiento muy grande de microalgas, y pasados unos meses toda esa productividad, todo ese alimento va depositándose y va llegando a lo más profundo del mar. Y una vez llega es cuando muchas veces se produce gran tasa de reproducción de especies como las que estamos viendo que se alimentan del sedimento.
Bridge ROV. Hey Luke, we have an updated waypoint for you.
Great. See waypoint.
Thank you.
Por tanto, parece que hemos llegado a una zona particularmente productiva, es una zona prácticamente productiva en una época particularmente productiva. Además de muchísimas holoturias, luego tenemos muchísimos juveniles.
Se puede ver claramente que ya vamos descendiendo en profundidad hacia la zona más alta de nuestro track de buceo. Avanzando por esta especie de ladera o colina hacia arriba.
look, no, not anymore because we're on the top of it.
Eventually,
We kind of,
right?
yeah, turn right a little bit just to, yeah, let's observe this rock in this side. And just see the other side, just zoom in a little. Oh, let's zoom in into this one. Yeah, that guy.
Antípodo.
And it's nice to see. Oh. Now we can continue. Well, if we can get, yeah, if we can get a close up. That's beautiful. That's perfect.
Lo que se observa debajo de Peracarido son numerosos tubos de poliqueto de la familia de los serpúlidos, muchos de ellos abandonados, ¿sí? Y también briozoos. Un anfípodo, ¿cierto?
Sí.
No hay
we
tal
can continue going
la
up.
capacidad de nadar de los ledmogones.
Parece que una esponja muerta.
Una esponja muerta así, gigante.
Era grande, sí.
Hay que destacar que las esponjas pueden llegar a vivir muchísimos años, miles de años de hecho se ha registrado. Por tanto son animales que dan cuenta también de la estabilidad también. Son ecosistemas, diría yo, frágiles pero también muy estables porque han estado mucho tiempo aquí.
Just to see who it is.
Ahí se ve una loturia nadando. Se ven dos isópodos abajo del grupo de los cerólidos que
No, yo solo veo uno.
se ven. Creo que el otro o es una marca.
Ah, una marca. Okay, we're gonna.
No sé si es una marca o está cubierto, el del más a la izquierda.
Sí, yo creo que son poliquetos de todas formas. Ahora lo
Ah,
miramos.
tienes razón, sí.
Ahora
Son
zoom
poliquetos.
in.
Sí, son poliquetos. Perdón el error. Son
Polinoides,
son
¿cierto?
poliquetos polinoides de los que tienen élitros o escamas.
We're gonna slurp that one.
Ah.
We're gonna slurp. We're gonna sample that one in this section. It's a beautiful one.
That would be great.
Poliqueto de aproximadamente 10 centímetros.
Control stuff. Copy that.
Thank you.
Y en este momento va a ser
We'll zoom into this one.
muestreado.
I'm not sure if there's another one or just a mark. We zoom in here or no here? Well, I'm not sure if there's several only one. Don't know if it's a. No, that's a fish, nothing. Never mind. But let's try to see if there's another one there. It's just. Yeah, there's another one. So we're gonna section both of them.
Son dos, claro.
Which chart?
Can be 7.
You can't get it there? Too hard? That's helpful. But if you can't get it, I'll try again.
Es decir, que estos animales muchas veces cuando se sienten amenazados sueltan los élitros. Son esas plaquitas redondas que tienen encima.
Sí, hay dos. Específicamente se ven dos poliquetos polinoides, muy próximos. Really, it's not a sea mouse. It's a similar species of kind of sea mouse, maybe, but it's different because the scales are bigger. Sea mouse belong to the family Aphroditidae.
It's in. I think it's in, but I'm not really sure.
Vamos a ver qué.
Section 7.
Section 7.
Anything else?
I don't think we're fine.
We what?
Paulite, pero es imposible sacarla sin hacerlo.
I'm just zoom into it. We are not going to collect because it's too difficult, but just to register the species. No? Yeah. Thank you. That's fine.
Algo así. Una estrella. Justo estábamos comentando sobre el animal que se veía sobre esa roca.
Que aunque parezca mentira, es posible que sea un equinodermo.
Sí, realmente algunas formas son muy difíciles de diferenciar a pesar de la espléndida imagen que tiene y el zoom que tiene las cámaras del ROV. A veces nos cuesta diferenciar de un grupo u otro dependiendo del animal. No es limpio.
Bridge ROV.
ROV bridge.
You can go ahead and continue to waypoint.
Continuing to waypoint.
Thank you.
Patri, fue un solus. Sí. Este es un solus. Era un solus.
Solo un tipo de holoturia bastante particular.
Sí, son holoturias que se anclan muy fuerte al sustrato y que prácticamente no se desplazan. Ya son holoturias irregulares, no con la típica forma de un pepino, digo.
Sí, realmente casi se comporta como una lapa. Es curioso siempre ver lo que llamamos convergencias adaptativas, que son animales o especies que provienen de grupos taxonómicos completamente dispares.
Claro.
Pero al final acaban teniendo morfologías semejantes.
Muy similares, sí.
Porque adoptan un estilo de vida semejante.
Eso ocurre mucho con el tipo de los vermiformes, gusanos. Muchos animales adoptan una forma de gusano en alguna etapa de su vida, porque es una forma muy óptima para en este caso, sobrevivir en estos ambientes. Ahora se ve claramente cómo estamos ascendiendo por este verdadero cerro o colina.
to these two guys. Just a close up. Never mind. Yeah. Yeah, I used to.
Como que estamos haciendo zoom, acercamiento a nuestros dos anfípodos más. Ahí hay un molusco.
¿Ah?
Claramente se ve un molusco. Corresponden a unas pequeñas lapas del género Yutia que se adhieren fuertemente al sustrato.
Es que si raspamos con el succionador un poquito
No, se va a quebrar. No conozco. Pero tengo.
Ya. We can continue.
Lo de siempre que apenas el ROV se acerca demasiado, el sedimento empieza a resuspenderse, pero cantidades enormes.
Are the... I cannot see them. Are the light... yeah, nothing. Never mind. I was wondering if the lasers are on, but now I saw them.
Bueno, mientras seguimos avanzando, voy a dejar el micrófono a mis compañeros para que hablen un poco sus impresiones sobre lo que es este buceo y sobre la campaña o lo que deciden comentar.
Of this cap coral. Esa close up.
Close up.
Yeah. Close, I want to see this. Yeah, great. Estos corales también llegan a ser relativamente longevos, ¿cierto? Lo que alcanza la madurez sexual como a los 15 años. We can continue. Thank you.
Hola, les habla Leslie. Nos encontramos a 435 metros con una temperatura de 1.17 grados. Lo que acabamos de observar es un cap coral, probablemente del género Flavellus, de las escleratinias. Seguimos subiendo por este paredón, donde podemos observar otros organismos como estrellas de mar, sea stars, sobreviviendo a estas condiciones tan inhóspitas, además de la variedad y la diversidad de la roca, que podemos ver que tiene cambios en el color y en la forma. Esta pared rocosa, a diferencia de la que pudimos observar en otros buceos, podemos ver un ecosistema totalmente diferente, con menor cantidad de esponjas, con menor cantidad de vida, pero sin embargo siempre está presente.
Thank you. zoom into this guy. It's cryptic here, but there he is.
Observa una de las especies de pulpo de acá de la Antártica. Está justo pegado en el canto de esa roca, adherido.
Por un interés, por otro tipo de interés, y en ese zoom es donde encontramos pequeños tesoros.
Sí.
Encontramos especies que no habíamos visto antes. Es una observación que está haciendo alguien, están haciendo en el chat, y es que en previos buceos, y de hecho decíamos que ocurre muy frecuentemente, que los erizos de mar... Can you look at this sponge, please?
Wow.
Decíamos que los erizos tienden a ponerse encima concha, por un motivo que no terminamos de entender, es bastante desconocido. Eso ocurre en todo el mundo y en todas las profundidades. Siempre hay especies de erizos que tienden a ponerse algo encima. Sin embargo, aquí por cualquier otro motivo que también desconocemos, es cierto que los erizos no parecen tan propensos a llevar ese sombrerito. ¿El por qué? Pues no lo sabemos. Can we zoom into the sponge, please? Yeah, wonderful. And to see this guy. Oh. Well, turn off your never mind. Yeah, just as zoom into the sponge, I want to see what is on top. La esponja, al ser tan grande y de una textura tan particular, sirve de refugio, de sustento, o simplemente de sustrato a muchas otras especies. En este caso tenemos una fibra, un pez. Frecuentemente hay anfípodos asociados a la esponja.
La especie de ofiura que se observa es probablemente del género Ophiacantha, especie vivípara o antártica. Alguna.
Okay. Let's have a look to this rock. I'm gonna try to. I would like to try to see the other side. If there is one. Let's try to go along. Yeah. continue to the next week.
Qué impresionante la cantidad de la misma especie de holoturia que se ve en esto. En el fondo se ve una esponja bastante grande.
Tenemos un pez que no se confirmado antes. You get a close up of the fish. Just zoom in.
Bridge.
Yeah, we can go and proceed to waypoint.
Yep, on the move.
Thank you.
That's fine, thank you.
Lo que se observa al lado es un lirio de mar de esta especie que Puede desprenderse del fondo y nadar. Crinoideos.
Just briefly, we've seen a few of them. They're always beautiful. Let's just we can look at it briefly and then continue.
Otra especie de las que también hemos visto en algunos museos atrás, que son estos hidroides solitarios de tamaño muy grande.
Hay un tenóforo ahí atrás.
Y un tenóforo también, sí.
Ya hemos visto estos hidroides un montón de veces, pero es tal la belleza que es difícil resistirse
Sí.
a mirarlos por un ratito. Wow. A close-up of the sponges. Esta roquita es interesante.
Pero es muy interesante por la cantidad también de poliquetos tunícolas que tiene. La esponja.
Un pequeño jardín, un pequeño bosque en miniatura
Sí.
de poliquetos.
O figuras.
Es muy interesante que muchas rocas estamos viendo la misma cantidad de poliquetos, pero tubos abandonados.
Y en este caso no.
Y en este caso no. Debe tener que ver con muchas cosas. La orientación, las corrientes, cómo estaban cambiando.
Seguramente, claro, como esta área estuvieron cubierta por hielo, o sea, tiempo más atrás, seguramente cambió la dinámica de las corrientes, cambió la dinámica del flujo de nutrientes, lo cual hizo que muchos cantos de roca quedaran expuestos a bajos regímenes, seguramente.
Uy. Es muy difícil de filmar estos poliquetos porque enseguida que te acercas se retraen.
Se retraen y con el sedimento que se resuspende.
Yeah. They're retracted, so we cannot really see them. There's this one's
Ahí hay
still
algo,
there.
hay dos.
That we can maybe watch. Es que tienen como ese penacho de seta y luego tienen como una pequeña campanita, parece, ¿no? Esa pequeña campanita, el mecanismo de cierre del tubo. Entonces, cuando el animal se retrae, utiliza esa campanita para tapar el tubo. como una pequeña puerta redonda.
Una especie de opérculo.
No, wait.
Ahí se retrajo, se
Yeah,
retrajeron.
yeah.
Sí.
Yeah, we can. It's okay.
You see this?
Yeah. Although I'm not sure what it is. I think it's a broken. No,
Un
it's a
pedúnculo
close up.
de,
If we can get a close up of this one on
solamente
the
un
tip.
pedúnculo de monocaulus.
No estoy segura si está retraído o roto.
No sé si se está regenerando.
O regenerando incluso. We can get a good view of this tip.
Al parecer sí, ¿ah?
Me parece que es una regeneración. ¿No? Sí, interesante en cualquier caso. That's good, thank you.
6, 4, and 3 are those surf chambers that we've collected in so far.
Yeah, correct. Let's have a look to the rock. Yeah, let's have a look to the rock in here.
I forgot to mark down the last ones. Sorry. I just forgot where it's been. Sure.
we zoom into this one? Right, okay. I'll also briefly zoom into this one. Wonderful. Wonderful, thank you. That's good, thanks.
It's true.
As I was saying, I'm picking it up.
Oh, good stuff. Head into which point?
No ice or trouble?
Huh?
No ice or icebergs or trouble?
No. Like the rest of the facility, like.
Yeah.
Cool, thank you. Challenger, how shallow does this come on this dive?
Son malísimos. Parece ser una esponja muerta. Evitar calamar nuevamente.
I'm gonna have a look to this rock. A little closer up of the anemones or actinias. Yeah. Thank you.
Carry on.
Yes, please. There's a squid over there. There you go.
Nuevamente nuestro amigo ahí.
Hitting the seafloor for some reason. Yeah, I don't know why he's doing that.
We're blinding him or something?
The what?
We're blinding him maybe?
Possibly. To be honest, I have no idea.
Seen that a couple of times here with different things. Usually it's not a problem.
It would be interesting to dig a little bit into the behavior of these animals and why they attack like that to the seafloor for no reason. Maybe you're right, that you're just blinding them and they get upset.
Maybe down is sort of a reflex for some reason, diving down.
Station 12.
And there it is again. Possibly the same one.
Yeah, there's one that's been following us a long time. I wonder if it's the same guy. What you doing, dude? You wanna be my friend?
Now he seems busy with something. Whatever it is. The one was just like really hitting the seafloor.
Just spat something out. You see that? Little
Yeah.
black thing? He just spat that out.
Yeah.
Maybe he's hunting and that's
fish.
food or something.
should be in. Once we pass these rocks, we're gonna try to take the push course.
Push course.
Yeah. Push course and Olothurians.
Push course and what?
And sea cucumbers.
Niskin before we do anything?
Well, I don't know.
No, no Niskin this time.
Think you're gonna find enough Holothurians around here?
Possibly. I hope so. Well,
Kind of a long shot.
yes. But we're picky. We need the big ones. The big guys, those two. Oh my god, there are so many.
What, do you just scoop these and put them in a bottle box or something?
That's a good question. I think this one's. I don't know. How do we take these ones? We scoop or suction?
Suction. Sí, pero por acá.
Sí.
Bueno, tiene mucho.
Sí, pero la pregunta es si está con suction o con...
Con suction, pero con mucha
Con
suavidad.
mucho cuidado. So we'll do with suction, but very gently.
Suction?
Yeah.
Okay. Into a suction jar.
Yeah. That'll be number 8. We shall collect 3 of the big ones.
What jar you want them in?
Number 8.
Jar 8?
Yeah.
My jar 8?
Bueno, vamos a realizar alguna
Sounds ready?
los estudios tróficos.
Yes, sir.
Oh, that was a good one.
Number
Let's see again. Yeah.
Sound effects?
This one.
Alright, now do you want me to move or do you want to just try and reach somebody?
Let's
You want one more?
reach one more,
See if
a
we can
big
reach
one.
more? I can hop too if we need.
Is that one okay?
I think so, yeah. Well, we can take the 2 of them just to make sure.
You don't make the sound, it's bad luck. There you go. Now we're back on track. Alright, so there's your jar completely packed full of
Big
sea
Olothurians.
cucumbers there. So
That was number 8.
we'll go ahead and stop
And
and
then
put that there.
we can advance a little bit for the push course.
Alright, so that's off into, what's a jar I can put this on that doesn't have anybody in it? Is there one? Empty.
Yeah, which one?
Is there an empty suction sampler I can put it on?
One, 2 and 5.
One, but no, but I don't want one. Don't put it in one.
No, I just want to put it so they can't float out. Like one that doesn't have
You want to
one
put
in
it
it.
Ah.
in empty
You
one?
know what I mean? Because they can float at the hole if it's like this.
Okay.
So one is okay? There's nothing in there?
Yeah, one is empty.
One is empty. Okay. Let's go into one. Alright, there's one. Okay. And here you said you want to move forward and do some push course, right?
Mhm. Yes, exactly.
Good portion. Only we have.
That's all the way. Any words flying out?
Yeah.
Your squid buddy's still over there messing around.
Yeah.
I think he's trying to communicate with us. I am a scientist. I was reincarnated as a squid.
There's actually 2 of them. There's another one.
He's suggesting the vacation to Pushkar.
Yeah, but if you touch it, buddy, we cannot sample you. There's another one over here, actually.
Another what? Squid?
So, yeah.
Is it?
Yeah.
Okay.
Oh, I think.
You want to go look over there? Or?
No.
Right, I am parked.
Okay.
And,
This is-
your lights okay? You need different lights? What you want?
You could get rid of that spotlight in this one, and that was a conference one. Yeah.
We have all of them, right? 20?
Yeah.
We're doing 20?
Yeah, all of them. There will
Down a little
be.
bit. Alright, just let me know what you need.
Are you doing any additional completely filled ones?
Yeah, there will be the 3 long ones, and 2 more will be full.
Okay.
Bueno, a partir de este momento vamos a comenzar con el muestreo de los push core, que son estos testigos o núcleos para distintos usos o mediciones que queremos hacer. Por eso también tantos push core que se observan, son 20 en realidad, pero se hacen poco porque se distribuyen entre los distintos grupos de investigación que están en este crucero. Por tanto, a cada grupo le corresponde, según requerimiento y según lo que se estableció en el proyecto, le corresponde una cantidad limitada de push core.
Little squiddles.
Qué curioso, comentamos el calamar que está aquí.
Jorge Land.
¿Hay 2? Justo hay 2, sí.
No, hay 3.
Hay 3. 3 calamares.
2 a la vista y 1 jugando con una pala.
Parece que estuvieran cazando, dice los calamares, puede ser.
Sí, es cierto que nos están haciendo la vida más difícil para tomar estos buscos.
Observamos en la imagen que una de las periogonia o restauradores se puso sobre la Bioluminescence 4, porque si quiere venir con nosotros.
Hay otra acercándose también con
su lado tan característico, su plantación.
No sabemos qué intenciones tiene. Y un crustáceo también encima de la biobox.
Sí.
Somos muy populares en esta zona.
Sí.
The squid's gotta be what, hunting or something, huh?
Es posible.
Doesn't seem like it's too low.
This, wait a minute, before you put this one. So these 4 are full?
Yeah.
Yeah, so this one should not be full. We need 5 full, which is the 2 long ones.
The 3 long ones,
Yeah, the 3
2
long ones,
regular
but 2,
ones.
yeah. So,
Yeah, so 2 short ones are full now.
Yeah.
2 long ones are full, and number 3
Right.
is the full one.
yeah, and all rest are full. Thank you.
So we're good, right?
That's good.
Mhm.
Bueno, lo que estaba explicándose es que no todos los corales necesitamos la misma profundidad de enterrado, digamos, dentro del sedimento, sino que más bien algunos necesitamos una porción de columna de agua que es importante para medir ciertos parámetros.
También para estudio de meiofauna, porque es importante asegurarnos de que incluimos las especies que viven justo encima del sedimento, incluso nadando en el agua que está justo encima del sedimento. Realmente nos hemos acostumbrado a ver estas holoturias nadando y ya ni nos sorprende, pero realmente decía que nos hemos acostumbrado a ver cómo nadan estas holoturias, pues y ya casi ni nos sorprende, pero tiene una elegancia, una belleza muy particular.
Sí. Y se nota que ocupa mucho la musculatura que tienen para poder desplazarse. Aunque hay que destacar que son más móviles en la columna de agua porque en el sedimento se mueven bastante despacio.
Y tenemos en el fondo al calamar Jorge haciendo de la suya. Ya que nos ha hecho el trabajo bastante complicado. Porque
Está prácticamente a veces entre medio de los push corks.
sí, si realmente queremos estudiar bien dónde viven los animales en las diferentes capas, si nos altera ese sedimento nos complica mucho el estudio. Realmente nos altera esas capas que queremos estudiar. Bueno, parece que uno de los calamares ha sido demasiado curioso. Se acercó demasiado a los propulsores del ROV y va a servir de comida, de alimento a otra especie.
What's that?
And that's good, just got introduced to our process.
Oh, shoot, we did? Suicide angel.
Yeah.
Well, I see his tentacle in the suction, I think. Oh, no. Yeah, no, you're right. Poor fellow.
Ah, no, espera, no. Me equivoqué.
No puesto nada.
Nada.
Geological school for the shallows. They talk.
They're being taken already. They're all. Ah, no, yes. One of the long ones.
So
For the shallow ones.
yeah.
It's the number 3.
We don't have them.
If possible.
Course, we're not gonna take it here.
Okay.
We'll just leave it.
That number 3 is the long one?
Yeah.
Yeah.
So we'll just leave that one. So much stuff running. all of them except for 3?
Yeah.
Is everybody happy there? Put them back?
Fortunately, when they normally want to get a picture.
Yeah. Which one of them?
Yeah, to get a picture of the fauna. With all of them.
What's your man looking out for something like this? Or squid?
That's the dead squid head moving.
It's the dead squid.
It's not dead.
Okay. Something like that?
Yeah.
Hey everybody, good morning. Just doing a quick watch change. We've got Matthew Lee coming in for Patricia.
Okay. You happy there?
And we've also got
Yes, thank you.
Zach and
Now we
Sunny.
can put them back.
ROV right now.
Do you want me to move anywhere or just stay here?
Let me get these 3, and then if we have to reposition, then we'll get
It's just mid-morning here on Falcor 2, and hope you're enjoying the stream so far. Passing it over now to Matthew Lee.
You can move. You can move.
move the camera.
Good listener.
Down with your
Yep.
I don't know. Because there's an arrow there now.
It all seems extra tricky, doesn't it? Morning. up a little closer after this. Is that right, Sonny?
Yeah.
Hop in it. That'll be okay.
Yeah, that's good.
Okay.
This is floppy.
Do you want me to fix it?
No, I fixed it. That's all right. There you go. Yeah. Good morning, everyone. This is dive 790 of SOBastian. We're still down in front of the George VI ice shelf. This is our last dive here. Today we're doing the shallow portion of the area. So we're currently at 300 and something meters. We're currently collecting push cores. These are sediment samples that will be used for analysing meiofauna, macrofauna. There'll also be samples collected for bacterial analysis, for biogeochemical analysis. 3 push cores will be used for fatty acid analysis, which is used for looking at trophic connections, food webs. And 5 of the push cores will be used by the geologists who will be looking at sediment horizons. What that tells them about what happened here in the past. My name's Dr. Matthew Lee. I'm from the Centro IMA, Universidad de los Lagos in Puerto Montt. Part of the group of Chilean scientists looking at the benthos in soft sediments. With me I have Dr. Americo Montiel and Dr. Christian Aldea from the University of Magallanes. We also work with Dr. Eulogio Soto from the University of Valparaiso. And we also have a couple of students on board with us, Sebastian Aguirre from the University of Valparaiso and Leslie Novoa from the University of Magallanes. Buenos días a todos. Aquí estamos. Esa inmersión 790 del ROV SuBastian. Estamos todavía frente a la plataforma de hielo de Jorge 6. Es la última inmersión aquí. Hoy día estamos haciendo muestreo en una zona un poco más somera que ayer. En este momento estamos tomando push core. Ese push core se usa para análisis de meiofauna, de macrofauna, y también sacamos muestras para análisis de la bacteria, de la biogeoquímica. 3 de los push core va a usar para análisis de ácido grasa, porque usa para examinar la red trófica, los redes de alimentación que hay en el sedimento. También hay 5 push core que se usan los geólogos, se miran los horizontes y cómo eso informar sobre qué ha ocurrido en el pasado aquí en la zona. Mi nombre es Dr. Matthew Lee, soy del Centro IMAR, Universidad de los Lagos, en Puerto Montt. Y soy parte del grupo de científicos chilenos que están mirando la diversidad y ecología de los fondos blandos aquí en Antártica. Tengo conmigo Dr. Americo Montiel y Dr. Christian Aldea, que son de la Universidad de Magallanes en Punta Arenas. Y también trabajamos con Dr. Eulogio Soto, que es de la Universidad de Valparaiso. Además, tenemos un par de estudiantes a bordo, Sebastián Aguirre, que está en la Universidad de Valparaiso, y Leslie Novoa, que está en la Universidad de Magallanes.
2. Should be up here on this set though, once it clears. I wish that rail for the 4K was longer sometimes.
Well, we want to collect that, but probably that's not the best way of doing it.
Oh, wow. You want to try and drop them in a bottle box?
Yeah, it's not dead.
Which one?
Any,
Is that
no,
Luminescence?
in 2B.
2B. There's nothing in 2 that's going to fly around, right?
No, there's a fish
It's
now,
not dead.
apparently.
It's kind of
Okay, here goes B.
But it looks very. And is that a head up on the script on the right side?
Okay.
Yeah, it's coming home.
That's squiddies. Yeah. That squiddy's looking quite lively.
Help this, are you good? So you try and brush them off?
Yeah.
It looks like it's just that one tentacle, you know? Right there.
Passive sticky.
Okay.
You might be able to kind of grab them with the lid too. You know what I mean? Scrape them off.
Pay attention please. Good morning everybody. Just for your awareness, the general alarm, fire alarm will be sounded in about 9 minutes time. This is for exercise purposes. exercise purposes only.
Bridge, ROV.
Hi there, Zach.
Hey, Cap, just to be clear, we're going to leave the 2 pilots in here and everybody else is going to go to the drill, and then we're not going to do any moves or anything till the drill's done. Is that right?
Yeah, understood. 2 pilots, yeah, that's fine. But there's a bunch of scientists also excused. I'm not sure if you have anybody with you in there now that needs to be down there for the drill, but yeah, that's required. That's fine also.
Okay.
But yeah, we can stay still, or you can give us a couple of moves as well. What do you want to do now? You got any moves planned?
Yeah, we're just going to keep going on until we call this drill, and then we need to stop, you know, but if the watch lead stays in here, then we'll keep going too.
Okay, let's plan to keep going. I think this is where we don't slow down on the science.
Okay. All right, I'll call.
All right, thanks, Zach.
That's the instruction we got.
So you're going to stay in here? You're
Yeah,
not going to
that's what we were told.
And everybody else back here too?
Yeah, I think so. Yeah. For those of you watching, you're wondering what we're doing with the dead squid. We're going to collect that for tissue and DNA analysis. So,
I'm going to drop it in.
yeah. La muestra de calamar muerto es para análisis de ADN y otra análisis de tejido. numbers like Pushko on the end of the front there?
In the very front.
Is that 12?
11.
11, okay. Yeah. Thank you.
So the two short ones that we pushed all the way is 5 and 8, right?
Yeah.
Which one do you see?
All these, there's lots of them, but they're all...
No, Penyagon is, yeah.
Is that alright?
Yeah,
I don't see many scribble there.
if you see any,
Uh-huh.
I think Patricia already did a few.
Not according to the...
I don't...
There's nothing written here. I mean, there's one, there's one sucked up. But
Oh, one, okay.
Yeah.
So need to do very gentle.
Mm-hmm.
And if you see a scotoplanus, just
That's well.
retrieve it.
Yeah. Okay.
Because the
Move forward.
mobility that I have
No,
with
no, little too
scotoplanus
far,
now
huh?
is low.
No.
Yesterday 3 were completely, I think you saw.
Mm.
Yeah. So if you see a scotoplanus, yeah, do it, or any other.
Yeah, okay.
Yeah?
Yeah. No, there's no shortage of scribbles for Right. Right. Thank you.
Anything else there?
What I'd like to do is just hop a few meters, get out of the disturbed area. We need to collect at least 3 sea pigs and close the nisking hole.
Okay. Sea pigs are not the ones we just did, the pigs. We just got 3 cucumbers
Oh, okay.
a minute ago into one of those suction sanders. That's
Oh,
what
okay.
you're talking
It's
about, right?
3, okay. It doesn't say 3, it just says
Or-
hollows. Yeah, okay.
There's that squid snoot if you want it.
No, I think we've got enough dead squid, I think. And in that case, can you just close the nisking bubble before we move too far away? So this is 23.
No nisking. Do 2.
No,
Yeah.
just get why not?
it's written.
There.
All right, 2 wet.
No, stop. Stop. Okay, 2 wet. Sorry. Teresa asked for a nisking bubble.
No? No nisking?
No, it's fine. It's done.
ROV bridge.
Go for RV.
Hey, good morning. Just to let you know, I have the con and we can just continue as normal during the drill.
Copy that, thanks. Okay,
Thank you.
so I think we're ready here. I'll do a port in.
We're at waypoint 12, right?
Going all the way.
On the bridge.
Yeah, we did get 3
Yeah,
sucked
okay,
up.
that I wasn't
You want
clear
to
on.
move 10
No, no.
back?
Yeah, we want the scrotal planids if we see them, but I don't see any there. Yeah.
Yeah, we got 3 of these guys for sure.
Yeah. Okay.
But I'll keep an eye for the other fellows.
Just.
Scrota peanut butter.
So I guess all that. Yeah, we just keep transiting and see what we see.
Oh wow, we just spooked something big over here. It's not me, believe it or not. Someone else did that.
We're not that deep. Maybe.
Let's see if we can see what it is. Looks like it's coming from the right. Pretty big beat up area of the seabed.
Maybe a whale came down to visit.
Wouldn't be the first time.
Yeah.
Wow, that's huge. Yeah, look at that. That's something just whacked this area. Okay. Anyway, head on. Weird. Better come back.
Oops, stop. Nice.
Oh, you scared the bejesus out of me there, man. Good
Suction,
Lord.
right?
Just make sure you scream next time really loud. Just gonna go change out my chair and then we'll be ready.
You don't.
Alright, so yeah, you want to suction this guy?
Oh, definitely. Yes, please.
Your trilobite?
So we can put him in number 5.
Make sure I'm parked here.
Yeah.
Alright. Kind of semi-steady. Now, index, you said 5, right?
5, please, yes.
Let's see. 8. 7. 6.
Oh.
Back the other way, alright. Ooh, okay. Okay, there we go. 8. 1. 2. 3. 4. 5. There we go. That's what you want, right?
Yes, thank you.
Okay. I'm ready. If you're ready, let me get more zoom here. You want zoom on both cameras or just the color? 4K.
Yeah. Section on.
Pretty.
Oh, take a picture of it first.
Yeah. It's a big one as well.
Yeah, he's cool looking. Good luck, sucker.
There he goes.
He might be too big.
Yeah, he's right in there in the middle.
Okay,
You
we'll
can
stick-
turn it where we can see him though.
We can stick him in the buy box then.
I'll do my light so you can see him there. You see his head sticking.
Yeah, I can see it, yeah. Okay, so drop him in the buy box then if it's...
Can he swim though? If we let go, will he swim?
Possibly, but...
Okay.
We had them in the buy boxes before. So.
Porch out?
Yeah, so we put it in buy box 4.
Box 4. unbend the end of our little suction deal too. Alright, I'm opening 4. Nothing in there, right?
Shouldn't be. There's nothing written down.
Looking down with this guy too. He's all the way open. And I'm ready to stop suction. Yeah?
Oh. Okay.
Okay. It's off. There it goes. You went in?
Yeah. Close it.
Doof.
Yes. Thank you.
Deal.
Are we gonna flip that back in? The other one?
We could, yeah, or could maybe on a rock or maybe on one of those handles. I don't know. You want to mess with it now, or you want to mess with it next time we need it?
Okay.
Next time. Coming out of the muck. Okay. You want to go straight to waypoint 13 or kind of cut around the corner here a little bit?
No, we've got plenty of time. We'll go to waypoint 13.
Bridge, ROV, new waypoint.
New waypoint.
Actually, can we have that starfish? Looks different.
You want it?
Yeah, I think we'll take it. We've got plenty of space. I haven't seen one like that myself.
We saw one yesterday, but I don't think we sampled it.
Yeah.
It was bigger.
And you saw the ones with the black tips?
No, I had it in the armpits like this.
Oh.
The black tips too. All right, landing now.
Or what do you want? Something?
I'd probably just pick it up and stick it in the bio box.
No problem.
So, Josen?
Yeah, please. More visible on this one than that. Yeah, bio box 4, please. Yeah, that was a bit too much muck. Yeah, we'll take the glamour shot on board.
No glamour shot.
Yeah, well.
And let's see, I've gone all the way in on my camera, and I'm going to porch out. Ready?
Yeah. 4A if you can.
Okay, open a 4. Okay. Other fellas down in there, huh? Old trilobite looking fella.
He's in B, hopefully. Join the party.
Okay.
Thank you.
Door's closed. Porch 10.
Okay, present. Exit.
Moving on. Big old wall here.
Let's have a wee look. Enemies. What's that on the top?
Sloop and astroturf, I think.
Check it out, make sure it's not.
260 meters.
Can we have a zoom on that?
Did I see spider in there too?
Looks like it. Oh, crap.
Park.
Oh, there's a big amphipod in there as well.
Big amphipod?
Amphipod just there.
It's probably hiding from the spider.
Ooh. Oh, yes.
I don't move, you can't see me.
That's some definite collection material there. Can we go for a suck up this amphipod first? Because I think that's the more likely to swim off. And then we'll get the sea spider.
Can they go on the same thing or no?
I think the sea spider's a bit big. Plus, it might actually be hunting the amphipod. So I'll put it in the same thing.
Do you need me to move to probably get over there a little closer, huh? For suction.
Yeah. Let me get before you move, let me get this one. Okay. Good position.
Okay, I'm just gonna hop over a little bit.
Yeah.
Meter or two.
Oh, you know, Captain.
Alright. Oh wait. No. Flop. Stay. Frame in. Which one?
Okay, stop there.
Alright, we're on 5, that's where it's going, is that right?
Yeah, that's fine. Yes.
Okay.
I think I should flip this lift up first.
Ah, yeah.
Nope.
No,
Either. I've never seen it do that before. Silly. A little bit silly. prey. Ready for suction, let me know.
Okay, you ready, Joe? Right?
Go.
Yeah,
5.
yeah, 5. Alright.
Your attention, please, for exit back.
On that branch.
There you go.
Oh,
I
there
think
it
that
goes.
was it.
That was him. Alright, I'm looking, and
We want the spider as well?
there he is. Okay, he's in there.
Do we want the spider as well?
We do, yeah, but
He probably wants him in a different jar, though.
hold on, I didn't see the arthropod come through.
Oh, I saw him.
There he is.
Yeah, he's
There
in.
he is. Yeah.
Alright, so what do you want now?
So,
There's 1 and 2 free. 1, 2, and 8. I think.
no, 8's full.
8.
Yeah.
Only 1 and 2?
Yeah, got to be 1 or 2.
Okay, come on through. Go.
Yeah.
Whoop. He went in there. And
There he
skidoo.
goes.
Right, now 1 doesn't have anything, right?
Yeah.
Alright, there you go. Now we're on 1.
Round back out of the starboard.
Ran back out with the starboard. Index.
So 2 we put down and
Yeah, 2.
is that another one there? You want that one as well?
Where?
Could you
No, I think that's just, it's an urchin. Urchin, yeah.
okay, first one's still
Okay. All good there, and we're happy to move along there.
Yes, thank you. Wee fishy.
Alright, I'm going to turn right. You can start moving the boat to the
okay.
south there if you want, quite a bit. Put them over there by 14.
Yeah. Bridge ROV, updated waypoint.
Updated waypoint.
tell them to speed up if you want.
Where's your RV? You could speed up to 0.4 if you want.
Yeah, we're making 0.4 already.
0.8 then. Like he's using knots.
If you like, we can go 0.6 or something.
Yeah, 0.6.
Yeah, because I think whatever he uses is half of that.
Speeding up to 0.6.
Copy.
Seafloor. We got a lot this morning. We need to climb into a lifeboat.
That's the way to do those drills right there.
I've done it enough times now.
Honestly, most boats you don't put in. That's kind of weird to hear.
Yeah.
You got a muster, but then you can't.
I guess you get a large chunk of the science bar who've never done it, and they need to understand it's uncomfortable.
Things like probably. Not seen
The sheep eggs you're talking about?
Yeah.
Yeah, that's it.
They don't look like they would survive much of a current though.
No.
Maybe that's it too.
Although, on the last, in the canyons cruise, in the landslides, on the vertical canyon walls, the landslides, hundreds of tiny little sea pigs everywhere.
Really? It was more like a sea pig slide.
Yeah, no, that was the only thing that was there. So obviously they're nimble enough to deal with that. The sediment's so sticky, I think it'll take some pretty major movement to get it to submerge. Look at that.
Yep, there's a big fan coming too. You see that?